This conference will now be recorded.
From field cultivation to storage, foodstuffs are heavily affected by pests that reduce yield, leading to severe food insecurity in most sensitive areas. Indeed, food security and shelf sufficiency are major concerns for people in Africa, where cereals and legumes are preferentially consumed and constitute a source of food for livestock and poultry. But because of the climatic hazard, they are generally grown once a year at a fixed time, whereas consumption is intended to be year-round. It is therefore important to control the quality and quantity of crops as well as their preservation. So insect plant diseases and weight are major constraints to food crops production. It's now becoming more difficult to control by conventional method because of pesticide resistance, product withdrawal from the market, and the persistence of pesticide residue in food. We then need to control the emerging threat by developing a sustainable pest control system that will be environmental welfare and can maintain crops quality, productivity, and then even the profitability. So that's why we decide to focus on biopesticide, which are any type of pesticide extracted or derived from natural materials. Generally, biopesticides fall into three major classes, and they have several advantages, like they are safe to use, they are cheap, they are renewable. Their use, they can be used in household for insect and repellent. They are non-toxic, and they are out of resistance. So we then interest ourselves in several plants, but anyway, the study I present here was focusing on the neem, with other name as Agaricus indica. It is a plant that has proven its extraordinary biological activity. So, and the plant is widely spread in Cameroon, especially in the northern part of the country, and it is traditionally used in the pharmacopoeia and in crop protection. The plant has been the subject of many scientific articles and several conferences confirming its extraordinary biological activities. So almost all the parts of the plant can be used and have beneficial properties. We can use the plant leaves, we can use the bark, the seed, the flowers. Let's note that the seeds the seeds are very rich in oil, so they produce an extraordinary oil that is really used in Cameroon. So for this study, we decide to focus on several parts of the plant. We work, and we decide also to work on several type of pests. So on this slide, you can see some pests destroying cereals. So as I said before, we use different parts of the plant. We use leaves, seeds, herbs. We use the oil cake, even the oil of the plant, as well as let me not forget, we also use the bark. So the different biological plant sample were to different group according to the treatment we wanted to apply. So for the powder material, for example, we had the control that we applied 1 gram up to 100 grams. And for the oil, we started from 0 microliter to up to 1,000 microliter. So not extend much on the methodology, but as a result, we obtained one different plan tested, maize, um, in this slide, this table shows us the induced mortality of different insects, but some animation. But you can see that on maize, for example, on the first pest was maize. The leaf powder applied at 100 grams considerably inhibited the pest growth. If you look at the cake powder, we also had a considerable inhibition of the growth of different pests. So this slide is presenting the inhibition or the cumulative percentage of some pests. The pest here is Callidea degree. So this other slide is the inhibition or the cumulative percentage of mortality. This one was in vitro of different parts like leaves, bags, and all on Aftona pp. So you excuse me, but I'll not be able to add more result. But from what we got, we discovered that there was significant positive effect that were obtained on the protection of plants in the field and during their storage. So NIM, with scientific name as a director indica, as insecticidal power, and that is efficiency with an efficiency depending on the part of the plant that is used. So the dose and the insect pest on the dose on an insect pest. So it's therefore an excellent alternative to dangerous synthetic insecticide. So we not be much longer. I will thank you for your kind attention. Thank you.
Thank you, Ms. Dr. Johu. It was a nice presentation. Is anyone having any questions? Okay, thank you. So we can move toward the next presentation, and it is from Dr. Shrikar Pant from BGSB
Thank you
University.
too.
Yeah, thank you. From BGSB University, India. Hello, Dr. Shrikar Pant. Are you ready for the presentation? Hello, Dr. Shrikar Pant.
Yes.
Yeah, are you ready for the presentation?
Yeah, I am ready. I am ready.
Yeah, I'm going to make you a presenter. So you
Okay.
can share your screen and start your
Definitely.
presentation.
Okay.
Yeah, it's done. Yeah, it's visible now.
It's visible now, but it's
Yeah,
not visible on my side. Okay, yeah, it's visible.
yeah, it's visible. You can start your presentation.
Yeah, just wait for a minute.
Okay.
Okay, it's visible.
Yeah, it's visible.
Okay. Thank you very much. And I am highly thankful. I am Dr. Shrikar Pan from Baba Ghulam Shah Badshah University, Rajouri, India, and thank you for giving me this opportunity. And I am highly thankful to the organizers for giving me such opportunity to present and showcase my work. Keeping in view the present scenario, that is COVID-19 pandemic, today's theme for the seminar or conference or webinar is directly or indirectly more relevant.
I'm not able to. I'm not able to see your screen. Sorry.
Yeah, you are not able to
Okay,
see
it's
my
okay
screen.
now. Thank you.
Okay. I am visible. My screen is visible.
Yes, sir.
Okay, and I am quite audible also, na?
You are, doctor.
Okay. Thank you. Keeping in view the present scenario, COVID-19 pandemic, today's theme for the webinar is directly or indirectly more relevant as far as the new opportunities or the role of the plant world is concerned. Further, my work can be an option for vocal for local or towards Atmanirbhar Bharat, a dream of our Honorable Prime Minister Shri Narendra Modi ji. My talk on dye-yielding plants of northwestern and western Himalaya, India, an untapped source for bioprospection. And I will be focusing on only two aspects. Number one is well-known dye-yielding plants, and number two, the less-known dye-yielding plants of northwestern Himalayan region. As we all know that the color is one of the main element of nature that made human life more aesthetic. Color are more directly and indirectly connected with our emotions, festivals, and qualities. In the past, humans used to decorate their surrounding with the help of natural colors that is obtained either from the plant, animal, and some other sources. Since prehistoric times, natural color obtained from indigo, pomegranate, jackfruit, manjistha, etc. are widely used for various purposes, either directly or in the combination of some others. Natural colorant and or dye stuff are an essential fraction of non-timber forest products that are widely used for coloring purposes in various industries. Nowadays, synthetic dye are used as a coloring agent, but due to their environmental, social, and health problems issues, now people are shifting from synthetic to natural coloring agents. Now the question comes, what is dye? Dye is a usually colored natural compound or blend that may be utilized for imparting color to a substance like cloth, paper, plastic, and others. Dye are one of the most imperative plant products as they are associated with our cultural beliefs, various religions, rituals, crafts, and also help in gratify personal personifications. Further, they do not have any side effects. And the art of the dyeing is old as the civilization of human civilization. And Ajanta Ellora caves. In addition to this, Ajanta Ellora cave wall painting showed effectiveness of dyeing craft that had been inherited from Indian ancient times. Atharveda also contained the account of natural dyes, and Bhrigu Samhita is also written by means of natural dyes. Color garment and the traces in the ruins of Mohenjo-daro and Harappa also indicated the presence of the dyeing elements. Bible has also mentioned the use of natural dyes, including the saffron. Further, the demand of natural dye or the green product is increasing day by day due to the worldwide public awareness and a number of less side effects in comparison to the other products. The period of classical age up to the medieval period acknowledged the tinctorial capacity obtained from the Indigofera tinctoria. The Mellotus philippinensis, Caesalpinia. Besides these are some black dyes were derived from the plant like Terminalia chibula, Umbilica species and Indigofera tinctoria. Further, the color fixation property of the alum was discovered during the medieval period and until the middle 19th century, all the fabrics were dyed with the use of natural products. Among these, indigo, Tyrian purple, longwood dyes are most common and very important. On the other hand, synthetic dyes. On the other hand, now the question comes, what is synthetic dyes? Synthetic dyes, which are obtained from the low-cost petroleum sources, generally have easy dyeing, superior fastness properties, wide acceptability or availability at a very low cost, and hence and have a large colors and the shadings. These dyes have received faster acceptability due to its easy in dyeing, reproducibility, and the cost factor. With the advent of the synthetic dyes, there was a rapid decline in the use of the natural dye and later was completely replaced by the former ones. And in spite of the being dangerous to human health, having noxious, allergic and carcinogenic effect, synthetic dyes have subjugated the market, sliding the natural dyes largely because of the tiresome extraction process from raw material to lower color value and the longer time of extraction. But on the other hand, the natural dyes are believed to be safer because of non-toxic, non-carcinogenic and biodegradable in nature. Moreover, India is to be considered a forerunner in the art of natural dyeing, and it has a rich floristic wealth of approximately 490,000 plant species, including 17,500 angiosperms. In India, nearly 450 plant species are known to yield dye, and over 2,000 pigments are synthesized from different parts of the plant. But ironically, only 50 taxa and 150 pigments have been exploited commercially. The Himalaya has been the perennial source of attraction and challenges to human intellect throughout the ages. In the Indian Himalayan region, the floral diversity was profusely used by the local folks for dyeing of wool since the decades. But the natural dye stuff manufacturers have concentrated only a few known plant species, and the use of the plant-based dyes for coloring at commercial level is to an extent at an initial stage, and the dye-yielding plant have not received much consideration or the considerable attention in comparison to the synthetic dyes. Therefore, the study was aimed to document and to discuss the knowledge on the known and newly source of dye-yielding plants of northwestern Himalaya, along with nativity, altitude, and some other important information. As far as the study area is concerned, for this, we selected northwestern and western Himalaya of Indian Himalayan region, which includes the part of Uttarakhand, Himachal Pradesh, Jammu and Kashmir. It occupies the most strategic position in the Indian Himalayan region with a wide range of topographic and climatic conditions from tropical to alpine zone, and supports a unique natural and socio-economic diverse biowealth. It has wide altitudinal range, right from 200 to 7,109 meters above mean sea level. The region is home a number of tribal groups like Bhotias, Tharu, Razi, Buksa, Jaunsari, especially in Uttarakhand, Gujjar, Gaddis, especially in Himachal Pradesh, Gujjar, Gaddis, Bakarwal and Pahadis, especially in Jammu and Kashmir. which are totally dependent on the various bioresources for their day-to-day sustenance. As far as the methodology is concerned, present work is based on thorough screening of available data and literature on dye-yielding plants, especially for northwestern Himalaya and western Himalaya. Literature has been extracted from the internet by using Google Scholar, by using keywords related to the topic. Further information on various aspects like altitude, life form, nativity was generated from the various published sources. Data was compiled and analyzed for the various parameters. Further, taxonomic validation was done with the help of plant list and IPNI, International Plant Name Index. Now come to the result. Plant has been utilized by man for different purposes since the time immemorial. And traditionally, a large number of plants have been used as a dye-producing plant or for dyeing in the Indian Himalayan region. Despite the high value of these chemicals, the high demand, slow availability, and very few studies have been conducted so far in this region. As far as the results are concerned, species diversity, a total of 87 dye-yielding plant species, 38 herbs, 25 trees, 19 shrub, and 5 climbers belonging to 45 families and 74 genera have been recorded. Leguminaceae is the most dominant family with 8 species. Just wait a minute, I think I have some problem. Okay, Leguminaceae is the dominant family with 8 species, followed by Asteraceae, Solanaceae, Amaranthacea, Balsaminaceae, Euphorbiaceae, Moraceae, Pinaceae, and Polyginaceae. As far as the altitudinal distribution is concerned, of the total species, maximum number of species, that is 48 species, occurs within the altitudinal range of 1001 to 2000 meters. Because this particular zone inhabited by the local inhabitant, mostly inhabited by the local inhabitant, that because of this particular reason, this zone is inhabited by the maximum number of species. Utilization pattern. The utilization pattern of the species indicated that leaves of 21 species, flowers of 18 species, fruits of 12 species, bark of 14 species, roots of 10 species, seeds, stems, and of 5 species were used for the extraction of dyes. As far as the nativity or representativeness are concerned, out of the total 87 plant species, 19 were native to the Indian Himalayan region, while 68 plant species were non-native. Most of the non-native species have their origin in the Asia, 38 species, while some other species were native to Africa and South America, Eurasia, and Europe and Australia. Out of 87 species, 27 species are well known as dye-yielding plants and exploited commercially. These are some in this Acacia catechu, saffron, Indigo ferretinctoria, Punica granatum, Tegetes, Rubia mangista, Melatolis philippensis, and this Impatiens balsamina are the common one, and rest 61 are also traditionally used for coloring purposes and on a very small scale, and very little information is available as far as their coloring properties are concerned, like Hibiscus, like Prunus armenica, Polygonium tinctorium, Whitfordia fruticosa, Pinus roxburghi, Pyrus perifolia, and Acacia fistula also. In this particular picture, you can have a look of the traditional extraction process, especially from the Indian Himalayan region, and it involves 3 major steps: extraction of coloring matter from the plant parts, creating a bond between the coloring matter and the fiber to be dyed, and third one is dyeing. this especially traditional wool dyeing from the Himachal Pradesh of Indian Himalayan region. Further, the synthetic dyes are harmful to both human health and the environment, and their harmful effects are very well known. Taking into account the side effects of the synthetic dyes, there is a worldwide drive in the use of synthetic dyes as a result of so many ecological, environmental, health and social problems. People are now moving towards natural dyes as they are eco-friendly and non-toxic in nature. Moreover, both the textile industry and the textile consumers are more and more aware of the need to develop and demand of eco-friendly dyeing methods for textile. Natural dyes offer a significant option since they are safer for use with at least least health hazards, easy disposal, biodegradable in nature, and residue can be effectively utilized for making compost for agricultural purposes after the extraction of dye. However, there are certain limitations of natural coloring dyes are as lack of availability of the sufficient materials due to complexity of collection, volume isolation of dye stuff or the bulk isolation of dye stuff is difficult and impossible. Extraction process is complex and has not yet been standardized. Limited color range or shades, low fastness properties. It is impossible to reproduce the same color or shade. Affinity of natural dye towards cloth is very poor. Therefore, moderant are needed to increase their affinity. Although the present study was limited to northwestern Himalaya and northwestern and the western Himalaya of the Indian Himalayan region, but its finding reveal that the urgent need to develop an appropriate approach and action plan for the optimum utilization of the availability of the dye stuff. Some principle in which we can have some principle upon which such a plan could be developed, such as, and it is necessary to document, it is necessary to have the complete documentation of the dye yielding plants together with their extraction procedure, availability and ethnobotanical practices should be carried out through the Indian Himalayan region. Sustainable method for collecting different parts of the plant should be developed and disseminated to the local inhabitants. Conservation and cultivation of the identified species will contribute to the continuous supply of the dye material, and the diverse rich areas or habitat need to be protected to ensure in situ conservation and continuous supply of the dye stuff. Identification and development of some alternative sources such as fungi and microbes through biotechnological tools. Use and promotion of the state art of technologies such as ultrasonic energy for the extraction that will save time as well as the plant bioresources. Now there is a need of education and awareness among the various stakeholders in the region for the conservation of these species. Thank you very much. Thank you for patient hearing. Stay home, stay safe, because we stay in, Corona stays out. Thank you very much.
Yeah, thank you, Dr. Shrikar. It was a nice
Thank you.
presentation.
Thank you
Is
very
anyone
much,
having any questions? Okay, as the next presentation is from Dr. Natale, but Dr. Randa has requested us that she will be giving presentation now as she has having some iftar on her timing. So, Dr. Natale, can we take permission from you to have a speech of Dr. Randa now?
Yes, okay, no problem.
Thank you, Dr. Natale. Hello, Dr. Randa. Are you ready to present your presentation?
Hi, how are you? Can you hear me?
Yeah, it's audible.
Yeah. Thank you so much for giving me the opportunity to present my work today.
Okay. So, Dr. Randa, I'm going to make you presenter. You can share your screen and start your presentation.
Okay, I'm waiting you.
Yeah, it's done. You can share your screen and start your presentation.
I'm trying to give the sharing,
Yeah,
but just
in
need
your
a minute.
screen, in the bottom there will be 4 buttons. One is mic, second one camera, and third one is screen.
I think I need some times because I need to shut down the application and then open it to share the screen. So if you
Okay.
go with the next speakers, sorry for that, but because my Mac say that.
Okay, so will it take time more than 5 minutes? Then we can wait, like if it is.
Okay. I think you go with because I will need to shut down the Mac actually to share. So
So
if you
shall
go
I
with the
proceed with next presentation? Till then you can prepare yours.
Okay.
Okay.
Okay, thank you.
Okay, thank you. Hello, Dr. Natale. Are you ready to present your speech now, as Dr. Randa is having some problem with his system?
Okay, yeah, I'm ready.
Yeah, thank you, Dr. Natale. So our next speech is from Dr. Natale Badlamenti, Pharmaceutical Sciences and Technology, Italy. So I'm going to make you the presenter now. You can share your screen and start your presentation.
Okay. It's impossible to share my...
Yeah, just a minute. I'm going to make you.
Okay.
Yeah, it's done.
Okay.
Yeah, it's visible. You can start your presentation.
Perfect. One moment. No. Okay. The name of my presentation is plant extracts such as insecticides. And the term pesticides covers a wide range of compounds, including insecticides, fungicides, herbicides, rodenticides, and others. And the advance of synthetic chemical insecticides introduced following the Second World War ushered a new paradigm in crop protection pest management. In fact, chlorinates, hydrocarbon, and other insecticides, also organophosphorus and carbamates, insecticides, were inexpensive to produce, relatively straightforward to apply, fast acting, and extremely cost-effective. They also offered remarkable flexibility for almost every pest. There were one or more chemical pesticides able to mitigate the problem. But organochlorine insecticides used in controlling a number of diseases such as malaria and typhus were banned or restricted after 1960s in most of technologically advanced countries. The introduction of synthetic insecticides such as organophosphates, carbamates, piterols, or the introduction of herbicides in the 1917 and 1918s contributed greatly to pest control and agricultural output. Ideally, but pesticides must be lethal to a target pest, but not to non-target species, including man. Unfortunately, this is not the case, so the controversy of the use and abuse of pesticides surfaced. In this slide, I report the use of insecticides in European member states, in general in the world, and a single case in India. As can be seen in the figure, in India 76 of pesticides is insecticides, as against 44% of the world. The main use of pesticides is for cotton crops, paddy and wheat. In terms of money, the demand of pesticides was estimated to be around $500 million dollars. But how do these pesticides act? Organochlorine, synthetic organic pesticides, are known for their high toxicity. Work on insects by opening the sodium ion channel in the nerve cell of insect, causing them to fire spontaneously. Thus, they inhibit membrane adenosine triphosphates, in particular sodium potassium, which plays a role in the repollination process. The insect will go into spasm and eventually die. Another insecticides, such as organophosphates, interfere with the enzymes acetylcholinesterase, disrupting nerve impulses and killing the insect. Normally, a nerve cell in the brain send tiny electrical pulses down tenderly to the end of the cell, where the pulse has a jump across a gap to another nerve cell. In this case, a chemical known as acetylcholinesterase moves from one cell to the other cell, sending the electrical pulse down the new cell. These insecticides prevent the acetylcholinesterase from coming loose from the new cell, so it can't receive any more impulse. The insect can't function and eventually die. Carbamates derived from carbamic acid are more degradable than organophosphates with lower dermal toxicity. For example, carbonfuran, a typical skeleton of carbamates, is a very up-to-date substance in toxicology, and birds are 10 times more sensitive to carbonfuran than mammals. Carbamates have similar properties to the organophosphates, but last in the environment for a much short time periods. They are thought to be less toxic. There are some benefits of pesticides, in particular in improving productivity, derived from the use of pesticides in forestry, public health, in the domestic sphere, and in agriculture. For example, in India, where food grain production, which stood at 15 million tons in 1948, had increased almost 200 million tons to the end of 1996. Similarly, for wheat yields in the United Kingdom or corn yields in the USA. Control vector disease. Vector-borne diseases are most effectively tackled by killing the vector. Insecticides are often the only practical way to control the insect that spread deadly diseases such as malaria. In quality of food, in countries of the first world, it has been observed that a diet containing fresh fruit and vegetables for outweigh potential risk from eating very low residues of pesticides in group, and benefit in another area such as transport, sport complex, and building. In particular, the transport sector makes extensive use of pesticides, in particular herbicides. But there are also other pesticides with a direct impact on non-human. Which results in serious health implications to man and his environment, but by impact to food, soil contamination, effect on soil fertility and water contamination. The high-risk groups exposed to pesticides include production workers, formulators, players, mixture loaders, and agriculture farm workers. Natural plants produce an array of natural products, and many of these are thought to serve an ecological function for the plants producing them, serving to defend the plant from herbivores and pathogens. This is a foundation, the idea upon which many scientists have viewed wild plants as a valuable resource for new insecticides compounds. But thought hundreds of compounds isolated from plants have demonstrated bioactivity to one or more insects based on BSA in the lab. Feeding deterrence or larval growth inhibition is far more commonly seen than death of the insect tested. In this slide, I report different compounds isolated from several plants such as Annonaceae, Stemonaceae, Meliaceae, and Scrofulariaceae. And during my thesis period, I studied the Drima pancratium, a species belonging to the family of Aspergillus, present in North Africa, Italy, Malta, and Corsica. It's cultivated in the Mediterranean area and used as a cardiotonic and diuretic for the treatment of cardiac marasmus. Previous studies of Drima maritima, another Drima species, results in the isolation of cardiac glycosides, flavonoids, fatty acid, and some studies have shown that the metabolites of this plant inhibit the sodium-potassium adenosine triphosphates. Drima pancratium, it was investigated against Stegobium panicum adults used its methanol and butanol extract. Stegobium panicum, also known as bread and biscuit beetle, it's among the major pests for a wide variety of dry and durable stored agriculture products. And furthermore, it's reported as a pest of cultural heritage because it feeds also on wood, old painting, and books. The structure of the isolated compounds from Drima was determined by LCPL-MS, proton-carbon NMR, nuclear overall effects, and two-dimensional NMR spectra. Regards contact toxicity and repellent activity of 5 different solutions from 6 to 100 microgram ml of the methanol and butanol extract wasn't found to be a significant effect as evidenced by the 2K test. But feeding tests performed with dry dog food showed a significant mortality of Stegobium panicum adults with increasing dose. The dose from 6 to 25 micrograms per ml didn't show any important effect, but only the dose of 100 milligram ml of both extracts showed a significant effect compared to the water control. Comparing the results obtained on the two extracts, the methanolic extract quantitatively evidenced a higher mortality toxicity than the butanol extract. Also, essential oils exhibit insecticidal, acariciidal, and larvicidal properties. Not only act as a poison on the nervous system of the insect, but also can intervene in the cellular breathing either by inhibiting cellular oxidation to transfer interruption in the respiratory chain, or by asphyxiation to the formation of the impermeable film insulating insect from the air. In this case, it's very important the choice of analytical method because it can influence on the determination of essential oil properties, which are mainly divided into microatmosphere. In this case, a disk of essential oil impregnated filter paper is depositing in the descent of Petri dish without a direct contact with the insect. Another method is direct contact. In this case, essential oil can be directly deposited or sprayed on the insect. And finally, in ingestion methods. In this case, the insects are put in contact with grains impregnated by essential oil. I studied the property of essential oil of Rhodolphia sagetum, an annual plant commonly found in Silla field in Europe, Turkey, and North Africa. In Morocco, for example, it's cooked as a vegetable in several recipes, including couscous. In Sicily, it's now as finocchianito is consumed raw in salad and cooked. The hydrodistillation of essential oil gave an oil rich in monoterpenes hydrocarbons with alpha-phelandrene, beta-phelandrene, and terpinolene made up about 18% of the total composition. Oxygenated monoterpenes were present in lower amount, with piperitone oxide at 5.9%. The essential oil is tested with against 3 different insect species reported in Table 2. Concerning Culex quinquefasciatus, the essential oil show had considerable activity against the third sinister larva of the study species, with LD15 of 27.1 microliter per liter. Regarding Musca domestica, different toxicity was found on male and female flies, calculating LD15 of 10 and 15 microgram for adults, respectively. Based on LC19 value, estimated for less sensitive female, we will be able to achieve at least 19% adult mortality, approximately with a 15 or 20% of essential oil-based preparation. A good activity of essential oils was found significantly for Spodoptera littoralis. Its efficacy seems too good against this important pest. In conclusion, so many plants species produce substances that protect them by killing or repelling the insects that feed on them. For example, the present investigation on methanol butanol extract of Dreamia could be a potential source of natural product that can reduce the population of stored insects such as Stegombium panicum. Rhodolphia sagetum provides an effective essential oil which can be used for the development and subsequent production of the botanical insecticides against diverse and harmful insect species. These prospects are also enhanced by the possibility of growing the plant even in arid and rather dry areas. Thank you so much for your attention.
Thank you, Dr. Natale. It was a very nice presentation. Is anyone having any questions? Okay, so we can move towards the next presentation. Hello, Dr. Randa. Are you ready for your speech now?
Yes, I am ready. Could you just
Okay, I'm going to make you presenter. You can try that. Yeah, it's done. Is it possible for you to share your screen now?
Yes, just 2 minutes.
Okay. Are you facing any problem? Yeah, it's visible now.
Yeah, can you see the slide now?
Yeah, it's visible. You can start your presentation.
Yeah, thank you so much. Hi, everybody. My name is Dr. Randa Al-Qurashi, Associate Professor at King Faisal University in Kingdom of Saudi Arabia. And thank you so much for giving me the opportunity to present today my work, which is talk about antioxidant and antimicrobial activities of moringa leaves growth in Al-Hassa region and chicken burgers, which is I will present today. So in Saudi Arabia, the Saudi oasis of Al-Hassa, located in the middle of the desert in the kingdom, which is recently has entered the Guinness book for recording as the largest palm tree oasis in the world, which is over 3 billion palm tree produce of some of a lot of dates, for example, trees and others, fruit and vegetables. And this is, as you see, this is the life which I took myself in Al-Hassa places which I live in, and which is it has a lot of trees, which is presented a lot of fruit and vegetables in the middle of the deserts in Saudi Arabia. This is some type of fruits and vegetable which is growth locally, as you see it in Al-Hassa region. For example, they have more than 10 types of date growth in this place, and very different type of berries like blackberries and blueberries, and also moringa olivers leaves, which is growth everywhere in the city. So that's why I present today my study, which talk about moringa leaves, which because no any studies have been done in type of the moringa that growth in Al-Hassa region. So why it's the moringa? Because it has contained high number of, as previous study show that, for example, in India and in China and in other places in the world, they have shown it's high in antioxidant and bioactive compound, which is contained, and which can we help to use it as a natural preventions, chemical compound added to the food, which can increase the shelf life of the meat or meat products contain. The aim of our study, which is investigate antioxidant and bioactive compound in moringa leaves growth in Al-Hassa region, and to see their antimicrobial activities in chicken burger. So why we choose chicken burger? Actually, in Saudi Arabia, the chicken fast food consumption in Saudi estimate all over the world, actually, by 4 to 5 billion around the world restaurant. Food market in Saudi, or fast food markets in Saudi, increase every year, which has contains a lot of chicken, for example, chicken burgers. For example, in Saudi Arabia, which is one of the largest worldwide consumption of chicken, eating, for example, 47 kilogram of chicken meat per year per person at average. So that's why chicken burgers consumed in Saudi Arabia, it's have increased every year. So we choose chicken burger to add it as a natural food product like moringa or other bioactive compound to increase the shelf life of chicken burger was our main in this study. So our methodology, we used moringa growth in Al-Hassa, which use two type of moringa, which we use extract moringa. We extract it with 80% methanol, and moringa, whole moringa powder. We add it to different concentration. For example, we use 3 different concentration, 0.5 and 1% and 2%. And we tested in during different time point of 0 and 2 and 4 and 6 days of storage under a fridge condition, which is 4 degrees. After that, we in each time point we tested our total phenols in the chicken burger and flavonoid and antioxidant activities in the moringa leaf in the chicken burger. And also we measure pH at different type during the different storage time after treated with the moringa leaves. And we test also antimicrobial activities, which we use to test total microbial activity or total bacteria in the chicken. And then after we finish, we test the sensory evaluation to see if the customer or our panels, they accept the chicken burger we added with we added some moringa leaves on the chicken burger. So this is the preparation of chicken burger, as we said, at different type of or different concentration of moringa that we use in our study, as we present here. And we use control, as we say, control without any additive of moringa or anything. But we prepare chicken at the same procedure we done all the sample. And after that, we test bioactive compound. Our results have shown that our moringa, which is growth in Al-Hasa region, has contain high amount of phenolic compound. And this is, we detect so far 90 bioactive compound, which is present in the moringa leaves, which is the highest. And this is all the type of bioactive compound we get it on our moringa extract. Also, our study showing that chicken burger treated with moringa, either with extract or with powder, whole moringa powder, at the concentration of 0.5 or 1% or 2%, had significantly lower total plate of count of bacteria at the end of the storage period, which is 6 days, compared to the control. That means our moringa, our bioactive compound added to the chicken burger, that increased the shelf life of chicken burger compared to a control without adding any bioactive compound or any moringa leaves. So our study showing that moringa leaf have provide high antioxidant and antimicrobial benefit to the chicken burger during cold storage at 4 degrees. Also, our results indicate that either, as we said, extract or powder at concentration 1 or 2% had antimicrobial activity in chicken burger, which is increase the shelf life during the 6 days of storage. Also, moringa leaf, can we use it? We can use it as a natural plant material to extend our shelf life, or the shelf life for the meat and meat product, which provide the customers foods contain natural, artificial, natural additives for the consumer and for the manufacturing as well. To our knowledge, this is the first study has to examine the antioxidant activities and antimicrobial activities of moringa leaves growth in Al-Hasa place and applied to the chicken burger. As we said, has contained high in antioxidant, and we can use it as a natural antioxidant additives that are viable for the food industry and offer potential maybe health benefit or to the consumer. Our next step for the research, we're going to test the moringa to the animal. We're going to start with rat at the beginning, and then we're going to extend our study to do it in the human study in vivo. So our next step, we are going to do it in September. We're going to test these antioxidant or bioactive compound from moringa leaves growth in Al-Hasa, if there is any beneficial effect to the rat, for example, reduce the cardiovascular disease, or after we collecting blood from the rat after a certain time of point, and then when we get a positive result, we can start with the human trials, which we test any beneficial effect of that antioxidant contained in the moringa to the human here in Saudi Arabia, or anti-obesity, for example, or reduce as antioxidant activities, or we can use it to see if there is any beneficial effect of the eating moringa to the human. So this is as presenting my my talk or my research I have been done during the last this year and last year with my team in Department of Food and Nutritional Science in Agriculture College in King Faisal University. In Saudi Arabia. This is my team. Thanks to everybody helped me on this study, and hopefully next month or after we publish this work to see, I mean, if everybody can see the results we got it in this project. So I will give thanks to everybody help me in this result or in this research, and thank you so much for all of you to give me the opportunity and to for your attention to this my talk, and welcome to any question, and if you want to contact me, this is my email and my Twitter. We can discuss or contact any further result or information you need. Thank you so much.
Thank you, Dr. Randa. It was a very nice presentation. Is anyone having any questions? Okay, so as we are ahead of time, so we can go to our next presentation. Dr. Diana, are you ready to give your presentation?
Well, I'm not doctor, I'm PhD student, but I'm here.
Oh, sorry, Miss Diana. Are you ready to give your presentation? As one of our speaker has not given their presentation, so your speech is ahead of the time.
Yes, there's no problem. I can start.
Okay. So our next speech is from Ms. Diana Nebeska, Gen Evangelista, Burkini University, Czech Republic. So I'm going to make you the presenter. You can share your screen and start your presentation.
Sure.
Yeah, it's done. You can share your screen. Yeah, it's visible now.
Presentation at the moment.
Yeah, you can start.
Okay, so hello everyone. As you heard, my name is Diana Nebeska, and today I will present results of our team, which is from cooperation from our Department of Environmental Chemistry and the Department of Biology of our university. I will talk about plant, which is called Miscanthus giganteus. It was also a point of presentation of my colleague, Dr. Al-Suki, who presented yesterday. So if you heard his presentation, I will repeat him a little bit, but just to remind, it's a perennial grass, which is one of the second generation biofuel crops. It is quite interesting because it can produce a large amount of biomass. It has C4 metabolism, so it has quite effective photosynthesis, low nutrient requirements, so it is available for production in marginal sites. And there are many ways how we can use this plant, not only for fuel, for heat, but also as excellent animal bedding, as mulch for plants, part of construction materials or packaging materials, or for production of some chemicals like bioethanol. But it has also great environmental profits. For example, organic matter sequestration or carbon sequestration. Karim was talking about it yesterday. Then support or the support of development of cell microorganisms. And it has a quite strong root ball, so it is great in erosion prevention. It's also because it's a perennial grass. And there's also application in phytoremediation, and it's quite connected to the topic which I will talk about today. And the inspiration for the experiment, which is part of my today's talk, was the pot experiment which we had in year 2017, which was with two different post-military sites. One was here in the Czech Republic. This is the one called Hradčany. It is a former military airport, and it is typical with very poor sand soil with a low content of all elements. And the other one was the one called Dolina, which is located in Ukraine, and there was quite elevated content of some heavy metals, and it was a soil with much better nutrition than Hradčany. And we found some interesting phenomenon which we did not expect, and it was that in content of zinc and manganese, the higher concentration in some plant parts was found in the poor soil, which had lower content of these elements. So we were thinking, how is this possible? And one theory was that this was caused by some sort of stress affecting the plant, probably nutritious stress. And it was also supported by our results from measuring of physiological response of plants which were cultivated in the poor soil. In these pictures, you can see always the green line represents a control plant which is grown in high nutritious compost, and the orange and blue curve are representing the plants growing in that poor soil, and you can see high differences, and we were able to determine some significant stress. But still, this was then quite surprising finding, and these experiments were not doing in the same location, so it was not so easy to verify the stress. Also, we didn't measure these parameters in Dolina, so we couldn't compare this physiological response. Then I've put these ideas together and prepared one experiment with large containers where we put these two groups of soils together. So we had two variants with poor sand soils, the same which we used in the previous experiment. So it was from the Hradčany airport, the post-military site. The next variant called C in this experiment was control. It was compost which was produced in composting plant, so it should be substrate. RHS, we measured it, which provided enough nutrition and optimal content of all elements. And then we had 4 variants which were mixed of control soil and contaminated soil, which was affected by high content of some heavy metals. It is a locality which was in history affected by smelting industry and ore mining. If you can look at the map which I borrowed from publication of Mr. Famněra, it is a locality here in the Czech Republic, and all of these small less like dots represent historic smelters or abandoned mines which are below the Litavka river. You can see here, and this river like flowed contamination, and it was settled and concentrated in sediments which we sampled and used. So we took the soil directly on the shore of this river. And here's a table with elements content. Those which are marked with red color are those which are over legislation limit which we have as permissible here in Czech Republic. So you can see that for all soils with some addition of or some ratio of contaminated soil, there was some increased level of arsenic, cadmium, plumbum, and also zinc. All these elements which we measured are those which are signed as risk elements in our legislation, and as a bonus we added manganese because we were interested in it because of the previous results. And here are the first reaction of the plants, which is a biomass. You can see that there's an obvious difference between the poor soil, control, and contaminated soil. And again, we found that the lowest biomass one was in the poor soil, while in the soil which was contaminated, the plant could grow quite well. And there was quite a significant difference between first year and second year, more importantly in control and heavy metals. We were actually quite surprised that it was able to grow so well in such contaminated soil. And it can be seen also in the pictures that the plant which was grown in poor soil called D2 doesn't produce only lower biomass, but it has a lower number of stems. It is generally weaker. Even it is quite high. But we didn't look just for biomass. Same as in previous article, we looked also in some physiological response. And probably most of you know the parameter of chlorophyll fluorescence, which can be quite nicely used for observing some changes in photochemistry, because when the leaf absorbs the light energy, it can be dissipated as a heat, use it for processes and photosynthesis, or radiated as chlorophyll fluorescence. So if there is some change in photochemistry, we can also observe changes in chlorophyll fluorescence. So it's a good parameter to look for a plant stress in general. For that, we use these handheld devices, which allow us to measure the fluorescence by non-invasive way. So we can monitor the plants without any disruption. We don't need to cut the leaves. And the process is that we put the plant in a dark using these clips. Then we'll wait for like 15 minutes. And then there's a light pulse, which like awakens the plant, and then we measure the reaction of fluorescence. This is a curve which is like dependence on fluorescence on time, and based on it's so-called OGIP curve, because like these steps are named by O, G, I, and P. And based on the shape of this curve, we can look for some changes in photochemistry. And generally, if the curve is like lower, like this one, it means more significant stress. So if we look for a green one, which is control here, we can clearly see that the plants which were grown in poor soil were much more stressed, but also the one which was grown in the soil with most contamination. But surprisingly, those two which were invariant with the lowest ratios of contamination were even higher in control. And we measured repeatedly during the season because we wanted to see also some seasonal variation, but generally the results were quite the same. We can use the same measuring also for calculation of different fluorescence indexes, which can give us some additional information, for example, for dissipation of electrons, electron transport, one so-called performance index. I will not go so deep into this because I'm primarily chemist, but generally we can look for another differences. For example, if you look at this parameter, it will give us practically the same information as we got from OGIP curves, that always the green line represents the control. These curves are normalized, so the control is always the value 1, and the other ones are relatively calculated to control. And again, the highest difference is between the variants with the low content of elements in sand soil and with the most contamination. We use also other approach, and it's measuring of leaf pigments, which can be traditionally measured by extraction of these pigments, but again we use a method which is non-invasive, and it's measuring of spectral reflectance of leaves, which give us again some different indexes which we can then compare and use for interpretation. Some of them are shown in here. The first one, so-called normalized difference vegetation index, is, let's say, some overall index of like green as soil vitality. We can see some differences again. Obviously, the sand poor soils are in worse shape than the contaminated ones, and the control is the best. The second one, MCurry, which is modified chlorophyll absorption in reflectance index, is closely connected to content of chlorophyll. We can see that there was a higher seasonal variation than in the first index, because the second measuring was done in October, which is like the end of vegetation season, when miscanthus slowly starts to lose its green color and turns into dry matter, which is then harvested for biomass. But again, these are obvious differences between the poor soils, control, and heavy metals. In this case, the higher value means higher stress, not like for NDVI. Another one is called so-called Carter index, give us practically the same information like this one, but not with such high differences. It's also some overall stress measuring. And the last one is carotenoids reflectance index, but same as for this one, which is attracted to chlorophyll. We can see some differences between those two measurings in summer and in autumn. But not so high differences between variants, but generally the trend is always the same, and it confirmed the previous measurings. But now we will shift from like stress and plant physiology to accumulation of elements. And this graph is mainly to show you the difference between uptake of essential metals and like toxic or heavy metals. Because even if we know that the concentration of some of these heavy metals was quite high in some soils, you can clearly see that the plants absorbs only the essential metals in some high concentrations, but it's known excluder of those like risk or dangerous heavy metals, which is quite beneficial because even if we grow this plant in contaminated soil, we can harvest quite clean biomass, which is great. We know that if we would measure the content in roots, which we did not do yet, we are going to do it in next month, the concentration would be higher because miscanthus is known for phytostabilization effect, which means that it accumulates the plants into its roots and it stays there. It's not transported to the above ground parts. But the content in biomass is always dependent on the content in soil. So for this purpose, quite more interesting is look for so-called bioconcentration factor, which is a ratio of content of these elements in plant tissues and content in soil. And if you look where the green boxes are, we kind of confirmed the results which we surprisingly found in our previous spot experiment, that for essential metals like manganese or zinc, the highest accumulation was for the soils which has the lowest content of these metals, which was not confirmed for those risk elements, which was expected based on the previous result. So get to the conclusions. The main conclusion was in the title of my presentation, and it's that nutrients deficiency affects miscanthus physiology more significantly than metals contamination, which is important for us because we need to know the limitations of our phytotechnology. So we know that it is possible to grow this plant even in quite highly contaminated localities, but it is not so good for very nutrient poor soils, or at least we need to expect lower biomass. And it was confirmed that miscanthus is excluder of most of heavy metals, but essential metals, which are necessary for plant growth, are accumulated more intensively. And the highest bioconcentration factor for these essential metals, specifically manganese and zinc, was in the poor sand soil with the lowest content of these elements, much lower than in contaminated soils with high content of these elements. And the hypothesis is this was caused by nutrient stress, because also in literature you can find like increased accumulation of some metals under different stresses, like heat or salt stress, but we didn't find many informations about this behavior under nutritious stress. But quite recent we got a reference which cited our article. They found the same. So obviously this hypothesis is right, even if we did not look for mechanism yet. And that's all from me. Thank you for your kind attention.
Thank you, Diana Nebeska. It was a very nice presentation. Is anyone having any questions? Okay, we can move towards the next presentation. It is from Dr. Prince E. Norman, University of Ghana. Ghana. Hello, Dr. Prince. Are you ready for the presentation?
Hello.
Yeah, hello, Dr.
Hello.
Prince. Hello.
Yeah, good morning. How are you, Ma? Yes, I'm ready
I'm
for the
good.
presentation.
What about you?
Yeah, praise God. I'm doing well.
Yeah. Okay.
Yes, I'm ready for the
So,
presentation. Can I
yeah.
share my
So
screen
I'm
now?
going to
Or...
make you the presenter. You can share your
Okay.
screen and start your presentation.
Okay.
Yeah, it's done. Yeah, it's visible, doctor.
So, are you seeing the screen?
Yeah, it's visible.
Okay, it's
You can
visible,
start
right?
your presentation. Yeah.
Hello? Okay. Good morning, everyone. Good morning, everyone.
Yeah, good
Hello?
morning, Dr. Prince.
Good morning,
Yeah, it's
everyone. I'm
audible, Dr.
presenting
Prince.
on trait profiling of white guinea yam breeding lines using breeding values and genomic estimated values. As we can see, what you're seeing there on the board are the reproductive structure of yam. Yam actually is a polyploid crop, but we have dioecious type. I mean, those that are diploid that behave in the diploid manner, especially the Dioscorea rotundata, which is white yam. So on our far left here, these are the fruits. We have the fruits as a female flower, and then we have the male. We have the fruits formed either from open pollination or from pairwise crossing, and then we have the seeds, which are often flat, having wings. And then the picture also we are seeing here, the far right here, is just showing the smile that we'll put on various end users of elite varieties that are developed. So we can see marketers here, even this woman smiling because of a very good variety that she is selling, even to have to have our daily bread. So the outline of my presentation follows this one. The introduction, and then right through to the acknowledgement. We'll be sharing various things, interesting things regarding what we obtained even in our studies. Yam is native to West Africa. It's actually native to West Africa, especially the white yam. And it has enormous end uses. It's used as food, feed, and industrial purposes, like even in the pharmaceutical industry. It can be used as feed for animals and even food for people. And then it often brings food and income to over 300 million people across the globe. And of these, over 90% is actually being utilized. I mean, the West Africa or Africa actually is the global leader of yam production. And but the production of the crop has lots of bottlenecks. So I shall be actually talking on the genetic bottlenecks or some of the problems that we encounter even to enhance genetic gain in this crop. They range from, of course, genetic, biotic, and abiotic factors. And then, of course, from the biotic factors, we have a number of pests and diseases that actually limit the high yield or even the productivity of the crop. We have the yam anthracnose disease, the yam mosaic virus, and then we have nematodes. We have, of course, the mealybugs often again affect the productivity of the crop, even the aesthetic value of the crop. We have soft rot, dry rot, etc. And then again, of course, the plant is largely affected by environment. There are environmental factors that can affect the productivity of the crop. Of course, a growing depletion in soil fertility, also affects the crop. And then we might need to understand what actually trait values are, because that's actually what our topic or title is suggesting here. So trait profiling involves genetic analysis of the existing divergence among genotypes and determination of functional relationships in the measured genetic parameters. It has wide application or uses, like in recombinant linkage analysis, determination of combining ability. It helps us to really, of course, determine heritability and other genetic parameters. Helps us to identify suitable and complementary parents, especially for crossing. You might want to use parents that have complementary traits, that you want to have them combined even in the progeny that you are developing, or the ideotype you have in mind to develop in crop breeding. So it's also applicable in QTL mapping, association mapping, and of course, in the heritability of disease resistance, determination of genetic estimates of even breeding values and relationships, especially when now you're using molecular techniques to estimate the genome estimated breeding value. I want to have some great relationships and explain better, the underlying factors, responsible for the expression of those traits. It helps in the selection process, to match. You want to select at the end of the day varieties, elite varieties that will be acceptable in the market. So having a good trait profile of varieties developed will actually help that process. It increases adoption of future releases of the crop and even other crops. And then, because nowadays we know the trend is to have correct product profile, which will actually help for market penetration. And then, so against this background, it's good to have a good understanding of the genetic profiles, which will help breeders and a suitable breeding strategy for the genetic improvement of the crop. So the objective of this study was actually to determine molecular and phenotypic values of yam breeding lines used in crop improvement. Now, this experiment was carried out at IITA Ibadan Station, and then a 6x6 triple lattice design was used. 36 breeding lines were used for this study, and 200 grams each of the sets, which were obtained from wire yam. We are first pre-treated and pre-treated in mancozeb, and then kept under shade for an hour, and then planted in holes created on the crest of ridges at spatial arrangement of 1 by 1 meters. And then about 32 agronomic traits were collected in this study using the production guideline or the protocol developed by AS4 2016. Now the photos here you see are just showing us, like for the starch properties that we have determined in the lab there, and also here is just showing us the protein extraction in the action in the lab. So just showing a few of those photos. Now, so for the DNA analysis, what we need to know is that about 3 months after planting, young leaves, young healthy leaves, we are collected and lyophilized. And so there's lyophilized leaves. We are used even for the DNA extraction using the Labora et al. 1983 protocol for the DNA extraction. So after the DNA extraction, the quantity and concentration of the DNA was determined using agarose gel and nanodrop, according to the protocol by Nazi and Martinez, 1997. So 50 microliters of the DNA sample per clone was actually sent to Canberra, Australia for that sequencing. So we received raw HapMap file from that, and then we actually used the PL programming language in TASO, for the conversion of those raw HapMap file into variant call file. So of the 20,000 SNPs initially obtained, we had about 8,277 informative SNPs, which were actually used even for various analyses. Now, so essentially, the selection of the key traits followed this pattern. These 32 variables that were first obtained was analyzed, in GenStat, of course, subjected to so that we can obtain the breeding value. So the blue analysis was done, and then identification and selection of key traits we are done actually using the principal component analysis, and then we have the variance inflation factor using the multicollinearity, and then the path coefficient analysis. And then for the principal component analysis, eigenvalue, of one or more than one, and the eigenvector value of 0.45 were actually used, as those components that actually significantly contributed to the variability, So using those cutoff points, of course, in the results, we shall see that about 31 still were retained, but we needed to have, key traits still obtained, so we also had to subject them to the variance inflation factor using the multicollinearity technique, and then variance inflation factor that are greater than one. Of course, we are less than one. From, don't mind this thing, it should be less than 10. We are considered in the first place, to have the trace reduced according to Habali 2020. But again, we then had to 14 key traits were obtained, but we have to further subject them, these 14 key traits, to path coefficient analysis, where we had yield as a dependent variable, and then the remaining 13 as an independent variable. And then, of course, when we used that one, we had all of them still below 5, giving below 5 values of the variance inflation factor. Now, after that, we also did correlation coefficient, just confirm, the view that traits with small and non-significant multicollinearity, should exhibit a low correlation. So those 14 key traits we are subjected to correlation coefficient analysis using factor minor in R. And then the statistical significance was done using the guide provided by Ratner 2009, which states that of course, if we have a value from 0 to 0.3, then the correlation is actually low. From 0.3 to 0.7, it is intermediate. the above 0.7, it is high. So those values still stand on the positive side. Then on the negative side, it indicates that it's negatively low. That's for those that are from from 0 to minus 0.3. Those that are from minus 0.3 to minus 0.7. That's intermediately negative. Then those that are higher than that, of course, they are strongly negatively correlated. So we also subjected those 14 key traits to genotype by trait by blood analysis, to see how the genotype and the traits interacted, so that those results also could speak volume and guide selection process. So those traits we are further subjected to prediction analysis. So using the single trial analysis based on multivariate mixed model using R test library, according to that author, we use that technique, and then for the previous one, the breeding values that we are modeled, we are modeled in G matrix to extract, so that we could be able to extract the genomic estimated breeding value, and then correlations with raw BV also for each trait to estimate the prediction accuracy. So we are able to have prediction accuracy and also the reliability of the results that are obtained from those. And of course, using those marker technique, we are also able to estimate the narrow sense heritability. So the broad sense heritability from the earlier technique, that's the breeding value, was also estimated, but that one was estimated on the mean entry basis, according to Robson. And then also the narrow sense heritability was estimated from the ridge regression blob model, and which took into effect, of course, a SNP effect. So we are now at results and discussion, looking essentially at this one, up to 10 principal components that actually exhibited, eigenvalue of 1 point. We stated our condition for selection of those that it has to be over 1.0 value. So up to here, and beyond this one, of course, it fell below 1. So essentially, we can see that up to 31, if you look at all the traits that are important or significantly contributed to the variability within the 36 clones, up to 31, we are retained, but we had to do further analysis. We can see that the principal component exhibited the largest contribution to the variability in the trait set, and the cumulative percent is about 80.34%. So we proceeded with the variance inflation factor, multicollinearity analysis, and then for here we could see that 14 key traits that had VIF value below 10 were selected at this preliminary stage, and then also these ones were further subjected to path coefficient analysis, in which we can see that the highest value here is just 3.1, which is the tasting temperature, So we can see that these key traits are really, really important. For instance, like for the dry matter content, which actually is very helpful, especially when you are developing yam, not only for food consumption, but now if you want to process it into other things, dry matter is very, very important. And then again, of course, the pasting temperature and all that gives us an indication of the, the starch property of the thing, and it's very, very important, you know, in breeding, especially for yam. Then of course the ash content gives us the mineral load, an idea of the mineral load, even in the various yams that are developed. So that one also is very, very important. Of course, the disease situation like yam mosaic virus, you know, very important, you know, especially in white yam. Yam mosaic virus is really, really an issue. So the area under disease progress curve was obtained by collecting, you know, the severity score, which are expressed in percentages over time from 3 months after planting. Let's start, I mean, from 60 days, that's 2 months after planting, right through to 180 days. So we used a formula that was suggested by Forbes et al. 2014 to estimate the area under disease progress curve.
Yeah, hello, Dr. Prince. Sorry for the interruption. Your voice is not audible.
Now, so we proceeded with the correlation analysis. We can see here that most of the associations were like mild, really low. Even things called by straight and exhibitions are more. Now, when we look at the genotype by tribe, the 36, there are certain associations with the clones. You know, instance, okay, for this first quadrant, we can see that about 9, you know, varieties associated with dry matter content, and then also for the leaf chlorophyll content. And we can see that TDR 99, 024, 74 is actually a premium genotypes among genotypes that had over 20 tons, you know, of yam, you know, per hectare, produce over 20 tons per hectare. And then also when we look at this other quadrant here, we have about 8 genotypes that are associated with ash content, the tuber shape, and then also, of course, the yield. Also, we had TDR 95 18544 and TDR 08/21 to also, of course, which are among the 5 genotypes that had yield, you know. For the TDR 89 02475 initial actually also with high dry matter content or tuber dry matter content. Then also we can see that here a number of traits associated with more, a number of genotypes associated with more, you know, about 7 genotypes associated with the base time, the pasting temperature, yam mosaic virus severity attack, and then the presence of spines above the base of the vine. Now we can see about 7 genotypes. And of course, again, TDR 9700917 was actually, you know, among great genotypes, you know, for some of these traits, especially for response to yam tuber mosaic virus, you know, protein content. Then the remaining genotypes associated with tuber flesh color, the stem diameter per plant, and the attack of Scutellonema bredis nematodes. Now, so for the heritability, both the heritability based on a mean entry basis, and then for the narrow sense, we can see that about seven trace had high heritability estimate. And normally high heritability estimate, especially again when it's combined with genetic advance, is a good indication of directing breeders to select genotypes, for those traits, and then to breed. It means that yes, those traits can be really heritable, and important to improve
hello,
the breeding
Dr.
efficiency for those traits.
Hello, Dr. Prince. Sorry for the interruption. Your voice is not audible. Hello, Dr. Prince. Your voice is not audible.
Just giving us an idea that marker-based relationship matrix enhances the capacity to estimate the narrow sense heritability based on additive genetic effects, rather than just using breeding value approach that captures a large proportion of the variance due to both additive and non-additive effects, resulting into inflation of the genetic gain. So we can say that actually using the marker approach is really good, and it's a good technique, to enhance breeding efficiency. Now, so the key findings are here. I will not repeat myself. Some of them I've lamented on already. So those are some of the key findings that we are obtained in this study. I don't just want to repeat myself. So the conclusions are that the young breeding lines possess useful variability and complementary trait values for young population improvement and selection for short release program. Also, breeding lines with higher breeding values for fresh storage yield, tuber yields that could be explored, could be utilized for young population improvement or recommendation for release. Some of those clones identified were TDR 0821/2. Then the others you can see for yourself. And then the genotypes with high dry matter content include these ones also that are highlighted here: TDR 04-219, TDR 89-021-57, and the rest also that you can read from the slide. Then those with lowest area under disease progress core value for young mosaic virus, they include these ones: the TDR 89/02475, TDR 95/18-988, and the others also that you can read from the slide. Now, so the recommendations from this study are genotypes with high peak viscosity breeding values for these genotypes, they exhibited, of course, high peak thing. They are still part of those conclusions, but now we are coming to the recommendation. Complementation of estimated breeding values with genomic estimated breeding values revealed higher prediction accuracy and genetic values of the profile white yam trace of progenitors. So we actually need to use this estimated breeding value approach so that it can, the genomic estimated breeding value approach, so that it can be able to help us, to really unearth the potential of those traits that we are looking for in the populations that we are developing. Is a good technique to complement the traditional estimated breeding value approach. Then useful key traits identified in this study could contribute to selection of superior breeding lines for both breeding and then recommendation for short release program. First, I want to thank the Coalesce Research Group for the opportunity to present in this great conference. An opportunity that I never treated lightly. Is really great. I want to thank God for my University of Ghana, and then the WACI program where I was trained to the PhD level. I'm so grateful to IITA, and then especially the African Yam program, and with whom I really, of course, did my research. I'm really so grateful to, of course, the government of Sierra Leone, and then to the Sierra Leone Agricultural Research Institute, for a great opportunity, always for me to share my research experience and work for the organization, and then for the IDRC for the support, they provided alongside with the Bill and Melinda Gates Foundation just to conduct this research. This is just a tinge of a whole range of great findings we obtained in that.
Thank you, Dr. Prince. It was a nice presentation. Now we can take a lunch break and we'll be back on 13:20.
Hello?
Yeah, hello, Dr. Prince.
Yes, I'm done.
Yeah, thank you,
Any
Dr. Prince.
questions?
It was a nice presentation. Is anyone having any questions? Okay, I think there are no questions. So we can take our lunch break now and we'll be back on 13:20.
Okay, thank you very much.
Yeah, hello. Welcome back, everyone. So now we can proceed with the another presentation from Dr. Patricia Romero Marco from International Osteoporosis Foundation and National Osteoporosis Foundation, Spain. Hello, Dr. Patricia. Are you ready to give the presentation?
Hello, everybody. Can you hear me?
Yeah, it's audible, Dr. Patricia. Welcome to the webinar. Now I'm going to make you the presenter. You can share your screen and start your presentation.
Okay, thank you.
Yeah, it's done, Dr. Patricia. You can share your screen.
Okay.
Yes, it's visible now. You can start your presentation.
Okay. How can I make this smaller?
Yeah, in the below you will be having a 4 icons on the screen. You can see the
oh,
mic, camera, and the screen. You can click on the screen. Or it's visible. You can start your presentation.
Okay. Well, let's do it like this then. So thank you for the opportunity to present my research. This is the starting point of the research, and we have continued later, but we still don't have the publications. So I will present some data, but approximately we will have more. Thank you for all the facilities of the administration of this Agri-Food Research Conference, and congratulations to all the speakers, because I think they've done very good research and presentations, and I've learned a lot, and I know much more things than I knew before. Thank you. So my research is about calcium and the adequacy of calcium intake among the Spanish population according age groups. First of all, I would like to introduce a little bit of calcium. And calcium is an essential mineral that must be provided by diet, and it's the only way we have as humans to have calcium in our body. Later on, we'll see how important it is to have enough quantity or enough intake of calcium. Calcium is also fifth of the most abundant elements, not only in human body, but also in earth crust and seawater. Here I have highlight two important things that are calcium. We can find it in human skeleton as calcium 40, and we will find also calcium with these two mobile free electrons as calcium 2 PET, because the differences are important as the benefits are different. So we have the first function or benefit of calcium in the body as the skeleton is structural, as calcium integrates the whole skeleton, the 206 bones that we have, and it's required to give to the bones and the skeleton the rigidity and flexibility and the strength needed to support the weight and all the movements. Also, it's important to know that it's a dynamic tissue, because bones are continuously forming and just turn over with their resorption. So we have we found very important to have the sufficient intake in all life cycle, but extremely important in children, adolescents, and elderly, and also within this menopause processes, because these formation rates are lower than the resorption, except in children, due to the growth, normal growth process. What we also find here is the bone marrow tissue, that it's associated with the immune system, and this is regulated by some calcium channels. So also calcium can be related somehow with the immune system. And the 1% rest is we found it in the intracellular as intracellular messenger, and these pathways that are very important. We will find out later that calcium is the major intracellular messenger for all the processes of the metabolism. As I presented, the calcium turnover, it's happening every day to everybody, and it's important to have the intake and the perfect and optimal gastrointestinal absorption, because we need to have this extracellular calcium in order to supply the needs of some vasodilatation and some other functions. If this extracellular calcium, serum calcium, is insufficient, the body will, osteoclasts will break bones in order to have to conduct them to the extracellular liquid. So we can fail on risk of osteoporosis or fractures or bone diseases. We can also have excess of calcium, and this is measured as the 2.75 or higher serum calcium. This is not happening, or it's unlikely to happen within diet calcium, unless we are taking too much vitamin D or supplements. Otherwise, the main reasons to have excess of calcium are malignant tumors or problems with hyperparathyroidism. So in Europe, bone-related diseases are a very big problem and a burden problem, that it will be increased within this 2030. And we can see here an osteoporotic bone and versus a normal bone, and also that the hip fractures and spinal fractures and other are the major places where we can find this risk of fracture. As I presented, bone-related diseases in Europe are very high prevalence. Approximately 20 million people have over 50 years old have osteoporosis in any of its grades, and we can see that as it's linked to this hormonal disorders or menopause processes, women we have 4 times more risk of osteoporosis than male. In Spain, we can see from this EU report that is expected to increase the osteoporosis risk by 29% from 2017 to 2030, and is the highest increment in the EU6 study in Europe. So we are observing that we in Spain we will have a high increment of osteoporosis, and this will have a very increment of cost and loss of quality of life of the populations. So to try to avoid this problem and to prevent this osteoporosis, Spanish local associations and also the European authority has developed or revised the recommended dietary intakes, and we have sorted them by age groups. We can find some differences between the Spanish and the European recommendations, because their recommendations are only indicative amount, as they are built as this is the calcium intake that should meet the individual needs of the 90% of the age group that are considered. So as we are considering Spain or Europe as a group, they can have differences. So this let's review, it's important, but because by evaluating general population calcium intakes and comparing them the findings to the recommended intakes, we can help countries or authorities to establish health policies and appropriate approaches to prevent bone-related diseases. So the aims of this LITS review are to examine the literature on the average calcium intake and of the general Spanish population, and to compare the findings of calcium intake with the Spanish and EFSA recommendation guidelines. So to carry it, we did a search from August to September in several databases, also including this Spanish warning dissemination network that it's mostly in the Spanish language. And we finally, after all the screening, selected 43 studies. The first results I would like to show that the studies designs were very different between them, and we could find very many types of designs as are here in this table. And another thing that will be also a limitation of the study is that calcium dietary assessment methods were also different in every study. We found 24 hours recall, and it was the most used methodology, however, not the only. Food frequency questionnaires that were, though all of them were validated, and that's a good thing. However, the food frequency questionnaires were not the same one, or did not have the same items number or in the questions. Food records, that they were the most used among children, and they all took one weekday, not 2 weeks day, and 1 weekend day. And the EU attendance that we found out that we also have several studies that they took the EU attendance, that it's a methodology that the European Authority developed in 2009. So we could, all of the countries follow the same methodology to unify the data. So going through the main results, we found out that as we can see, the here, infants and toddlers had very high intake, and they exceeded the recommended dietary intakes, not only the Spanish but also the European dietary intakes. We can find these big differences here in the 0 to 6 months because Dalmau et al. Took into account the breastfeeding kids and the calcium from the breastfeeding kids or the mixed feeding, not only breastfeeding but also bottle feeding. And however, Zaragoza Jordana, it was a PhD, and they had into account only bottle feeding. So this could be these big differences we can see in the data. In the other cases, 100% of the studies found out that children, infants, and toddlers reached and exceed the recommendation. So they are not in a risk of low intake, of low calcium intake. Children had also very high mean of calcium intake, and they also exceeded both European and Spanish recommended dietary intakes. However, when we moved to the adolescents, we could see that the mean calcium intake decreased dramatically, and this highlights a double problem, because from one part we have that these adolescents had less calcium intake, and the recommendations are the highest of the whole population because of the growing process and the bone peak formation that will be very important later in life when the menopause process. So none of the studies reach the recommendations, and not even 60% of the recommendations. Some of the studies were even less than 50%. The mean of calcium intake of some of the studies were less than 50% of the recommendations, which is very worrying, and it points a very high burden problem in Spain that we should manage or take into account. When we move to adulthood, the first part of the adulthood from 20 to 49 years old, we can see that even though the mean intake doesn't reach 100% of the recommended dietary intake, in the study we have calculated, you can see it if you read the paper, the mean intake is 80% or more than the recommended dietary intake. So adulthood seems not to have any problem or to be in a low risk. Something important are sampling that both genders are represented in this adulthood. However, when we move on to 50 to 59 years old, we can see that sampling covers mostly women. Well, this can make sense somehow, because this is the menopause age time. So it's kind of it's very important to researchers and also for populations, for women, to know how is the intake and the recommendations. What we can see is that from 50 to 59, women in Spain, mostly women, not only, but also, mostly, are covering the recommended intakes, also more than 80% of the recommendations, and the mean calcium intake is the highest found out in the whole population. So it seems that women within this age in Spain are conscious about this turnover, calcium turnover, and they are taking care of it, and that it's very good result. But if we move to elderly or more than 60 years old, then we have again the decrease of mean of calcium intake, and it doesn't cover even 60% of their dietary recommendations, which is a very big problem, because the turnover is also linked to age, and the resorption rate is a lot higher than formation rate. So elderly, it's also a point that we should take into account. About pregnancy, we have found 3 studies, and they covered almost 90% of the dietary recommendations, so they don't seem to have a problem. The limitations and the strengths, I've been talking about them, but we didn't account for supplements or vitamins or minerals. So this is a limitation of the study. We only took into account dietary calcium. The reproducibility is also a limitation because most of the studies were local or regional, and the designs and samplings were very different. So it's kind of difficult to see how to compare them. And another thing that it's important is that mean calcium intake does not report the distribution of intakes, and so we can not see the prevalence of people that are at risk of low calcium intake. And as a trend is that we are showing a global overview of the average calcium intake in the whole population, and we are highlighting age groups of population at risk of low calcium intake, so we can set the age groups that might be that can be beneficial from policies or health approaches to improve their calcium intake, and so their bone mass. As conclusions or key findings are that calcium intake is low in the elderly and adolescents in Spain, and that more investigation is, of course, needed. And I would like to thank you for listening, and please find down in the slide my email, and I will be very happy to collaborate with any of you if we can set any research together. Hello? Is there any question that I can answer?
Is anyone having any questions? I think there are no questions. Thank you, Dr. Patricia. Now we can go ahead for the next presentation from Dr. Ferizi, Secretary to the SILTA Western Australia Committee, Australia. Hello, Dr. Ferizi. Your voice is not audible.
Hello,
Sorry, Dr. Ferry, it's not audible.
can everybody hear me?
Yeah. Now it's audible. So are you ready to give the presentation?
Yes. Everybody.
Yeah. So I'm going to make you the presenter. You can share your screen and start your presentation. Yeah, it's done.
Hi.
Yeah, it's visible.
Everybody can see my slide.
But your voice is not audible.
Everybody can hear me? Hello. 1, 2, 3.
I think the volume is very low. No, Dr. Ferry, still not audible. Can I know, is it audible to anyone?
I don't hear nothing.
Yeah, it's not audible, Dr. Ferry.
We don't. Maybe you could try without the headphones. Maybe yeah.
Yeah, it's okay.
Yeah,
Thank you very much.
it's
So I'm
perfect.
not using the microphone, hopefully, so not having any noise on the background.
Okay.
Okay, just try to
Okay.
okay. All right. Good morning, good afternoon, and good evening. My name is Ferry J. Currently associate professor in supply chain and logistic management at Edith Cowan University in Western Australia. As you know, this is my details for my presentations. Currently, actually, I'm sitting under Chartered Institute Logistic Transport in Western Australia, as well as Supply Chain and Logistics Association as a steering committee. So what I would like to present today is about the future research in agribusiness and food, given my research expertise in a management and business area. So in pretty much about supply chain and logistics management. This is about my background. I currently publish more than 77 in a top tier international journal in the area of the business management, supply chain and logistic management, including food technology, as well as the doing a lot of research in halal food and red meat and beef supply chain, as well as a dairy supply chain. And also currently I received competitive international grants as well as the national competitive grant funded by Australian government from 2010 to 2020 is about 1.6 million Australian dollars. This is pretty much what currently my research grant. My research project currently dealing with COVID-19 in Western Australia, funded by Department of Job and Tourism, Science and Innovation, DGTSI, about the business resilience for capacity building and upskilling post-COVID-19 pandemic. So in Australia, we are a lucky country. Currently, we do not have any cases community transmission for COVID-19, so that's a good thing. So in Western Australia, pretty much, well, in the 2 weeks ago, we got 3 days lockdown. We got one community transmission, but so far so good. Every single day is 0 case. And only officers traveler in hotel quarantine still got the COVID-19 for about like 44 in the national for Australia. My second project currently just completed about logistic transport, funded by iMove Australia in Melbourne. And the third project just completed about small medium enterprises resilient, about blockchain, financial planning and advisory services supply chain. And the fourth project just finishing the report about waste, including food waste and food sustainability project. And then the second last completed before COVID-19 about logistics intermodal infrastructure for future need in Western Australia. I wanted to share something. This is very interesting about the FAO from United Nations Organization. I'm not quite sure whether we got time, but more than happy to share, send you the link about the scarcity of food due to COVID-19 situation published by United Nations Organization, FAO. And the second video presentation is about the COVID-19 global food security. This particular project is a very high demand in terms of COVID-19. I believe the project actually about global food security is a very, very important in terms of food safety, food security, dealing with the COVID-19 pandemic. As you know, the COVID-19 for the global, COVID-19 actually affected for the more than 220 countries around the world. So that particular research project is going on in terms of global food security. This particular, I would like to talk about theory of supply chain. I think most of you in this room, maybe you do not know the background of the business of supply chain. I just want to give you very basic fundamental of theory of supply chain. So businesses, companies, pretty much trying to design business model that fulfill the customer need better than competitors. It really depends on the ability, how do you design the particular product, you make a product and you deliver particular product to make sure they're innovative and high quality, low cost and services that customer demand. And then this is, I would like to share with you about supply chain. I've just give you one example about cereal manufacturer company, right? Everybody knows cereal product. So what does it mean supply chain in a cereal company? If you look at the upstream, right? And then you can see different tier. The first tier is a polyethylene bag manufacturer. We got flour processing, we got corrugated manufacturer. And the second tier, we got farmer, we got chemical processor. And the third tier, we got oil company, we got forest. And then you can see we have a downstream, which means that we have a grocery distributor, and we got retail, and we got consumer, and we got supermarket. You know, what we have here, cereal, pretty much from forest until we eat cereal like for the morning breakfast, you know. Pretty much is supply chain is a product and services, you know, coming from upstream to downstream, as well as information and planning. So that's why what is the purpose for today conference about agribusiness and food. This particular, I would like to introduce supply chain is very important in the food sector, in particular agribusiness. We looking at farmers, for example, and you know, producer, and then you have manufacturer, and then you have a retailer and consumer, and then we have a global export, you know, for instance, food product to other countries, right? And then if you look at the supply chain, is not only product but also services. For example, I give you one sector talking about hotel industry. Hotel industry is a focal point, but you got upstream, so you got labor provider, you got contractor for building, mattress, you got food services. And second tier, you got framing, cement contractor, and so forth. And then you got downstream supply chain, so pretty much hotel brand, and you got the business traveler and so forth. That is in product and services, pretty much from upstream, downstream. So supply chain is not only product movement, but also information and planning as well. So the definition about supply chain is the design of management of seamless value-added processes across organizations, boundaries to meet the real need of the final consumer. And I would like to share with you about Australia agribusiness and food. The Australia agribusiness and food sector is undergoing rapid change because of globalization. Highly competitive local export meat market, for instance. As you know, Australia, for red meat, we export more than 100 countries around the world. We export live cattle, we export processed beef, we export minced beef to at least 67 countries around the world. increased production efficiency, quicker production cycle, and delivery times are all contributing factors to this change. A trend towards more outsourcing of activity and the rapid development of applications dependent on information technology have also played a role in increased efficiency. That one particular I would like to share as well, the Australia meat industry. I published more than 8 papers about beef, Australia beef, Australia red meat, supply chain. Just want to give you the background about Australia red meat industry. So red meat includes kangaroo meat, beef, lamb, all red meat products. So we have the the Australia red meat industry is the 4th highest commodity export earner. We export about 65% of the production to over 100 countries around the world. In terms of the value productions, increased by $2.5 billion in the last 5 years. So, including during COVID-19. So we still, produce more for the red meat of beef, lamb, kangaroo meat, halal beef, and so forth. And it provides employment in terms of human resource, the workers, more than 55,000 employees right now. I just want to share, this is I developed the beef supply chain structure in Australia. So start from breeding property. So you got the feedlot, you got cell yard, you got backgrounding property, fattening, these yellow boxes pretty much upstream for beef supply chain. So, and then we got here processing, which is the focal firm, abattoir. And then we also process beef. We also export live animal, cattle, using sea freight, from Fremantle Port or from Port Melbourne. We export live animal to other countries. And we also have broker, wholesaler, and we import some particular product as well from other countries. And then we have food services, we have food processing, butcher shops, supermarket, and we have a domestic consumer. So this particular supply chain structure has been published in the Q1 Simago Journal article. So most of you can download and read my paper already published in the top tier journal about the beef supply chain in Australia. This particular framework I developed, beef supply chain performance. Supply chain, we really want to look at the business performance. What particular variables supply chain performance for Australia beef enterprises? We have a quality, efficiency, responsiveness, flexibility. Pretty much quality, I think most of you know about the food quality, right? Animal welfare and regulations and health, occupational safety and health in the workplace, environment, sustainability, skill, record all inspection and tests performed, operations costs, including waste costs, food waste, they're all about the efficiency. Responsiveness is about customer response. As you know, every country maybe has the same problem with the COVID-19. When particular city or lockdown, consumer panic buying, consumer going to the supermarket, they really want to get pasta, rice, any fresh produce, because lockdown is a consumer panic buying. It's just demand shock, because what happened, this is about supply chain. What happened if the demand shock is every customer want to purchase a food product in the supermarket? What happened if the supply problem? So no wonder we have a problem with the empty shelf, including if the person would like purchase eggs, for example, rice or pasta or noodles, yogurt or milk, even bakery or bread. No wonder, because consumer really scared they're going to be locked down, they go to the supermarket to grab all the food products And the next, maybe one day also, the supply problem, and the customer come to the shop, come to the supermarket, is empty shelf. This is what we call demand, uncertainty, which is what we call demand shock for the short term due to lockdown, due to COVID. As you know, a lot of producers, because COVID, what happened if the plant or food manufacturer, actually the worker got COVID, the manufacturer company gonna be shutting down, right? So this particular we can see about the demand shock. Now, this is about the competitive advantage in terms of the framework I developed for the Australian beef industry, looking at the price, food product, beef product, quality, of course, time to market, make sure the product is available in the market, make sure product still in a good condition, fresh quality, including if you look at the marketing and sales point of view, the business really want to look at sales growth. So this is the business really want to look at. The business want to look at the profit, they need to look at sales growth. As you know, profit is the sales revenue is minus cost. If you got sales is higher than cost, then the company really want to look at the profitability. It's pretty common in the competitive advantage framework for the Australian beef industry. That particular framework is already published in the International Journal of Logistic Management. British Food Journal is the Q1. It's a very, very good journal. Most of you, maybe you can publish in the British Food Journal. It's a very, very good, leading international journal. Okay, so in terms of list of my publication related to the food supply chain or beef, or as you know, those particular papers already published in the top 10 journal. Feel free to have a better understanding what is a food retail supply chain under Business Process Management Journal for the Australia context. And you can see I also work with the Indonesia scholar to look at the halal supplier service quality, and the project is funded by the Indonesian government. You know, halal food, it means that you produce food, particular beef or chicken, to follow Sharia law principle. It means Sharia law principle. Halal, it means permissible. It means that they have a particular rule in terms of Sharia law principle to look at the food process, make sure it's still halal condition. And that particular paper already published in the top tier journal. And the third paper, I think I mentioned before about Australia beef supply chain, published in the International Journal of Logistic Management. It's a very good paper, published more than like 5 or 6 years ago, about the Australia beef supply chain. And also, if you look at the next slide, this particular list of the papers are published with my doctorate student. Currently, I'm supervising 7 doctorate students. Most of my students actually publish with me together with the supervisor in terms of, for the research, and 2 particular papers now published with my doctoral student about halal accreditation, certification in a non-Muslim country setting, insight from Australia halal meat supply chain under International Journal of Supply Chain. And the other one is my PhD student as well, about institution forces on Australia halal meat supply chain under Journal of Islamic Marketing. This is a very good journal. And the third paper I published with my colleague from TU Berlin, about the dynamic recovery policy. This paper actually the case study about dairy, pretty much for milk, yogurt, cheese, ice cream, for dairy products in Australia. And these particular papers as well, pretty much published a few years ago, about the halal risk mitigation. That's project working together with Indonesia government about the risks halal for red meat product in Indonesia. And the second paper, the role of trust, antecedent cooperative behavior for the lamb retailers and wholesalers. So that particular product actually not only beef, I also focusing on a lamb, including kangaroo meat. Customer relationship strategy, cattle producers, which is live animal, and also Australia lamb processing industry. And then with my colleague from Chartered University, Professor Kevin Parton, published together with the need for improved skills in the Australian meat industry. Okay, so pretty much the list of publication currently, the previous grant I would like to share with you that I mentioned before, the effect of technological readiness of halal meat on halal meat logistic performance, funded by Ministry of Research, Technology and Higher Education of Republic of Indonesia. That paper actually also published as well just recently in the 2019, just before COVID. And then to continue working with industry, with the beef industry in Australia, we work together to adopt technology in order to support supply chain. That's what we call supply chain automation in the Australia beef industry. So what is the purpose of technology? Technology pretty much, if the customer or you got overseas markets, you want to place an order for the beef product, you can go to it, pretty much about e-marketplace or e-commerce. They can look at the beef specification and what level of the quality of the product, and the customer can place an order online, and they can negotiate in terms of the price, negotiate in terms of transportations, negotiate in terms of the what types of logistics service provider you're using it, in terms of the product, transport product from maybe from Indonesia, from Thailand, or from Malaysia, from China, Hong Kong, for instance, and whether the product actually coming from overseas, come to the Australia, or from or vice versa. That particular, we use a particular technology to support those processes. Now, talking about future research, so I think if you are interested in agribusiness and the food, of course, actually that's one particular important area or discipline in the supply chain of logistics, especially if you look at COVID-19. So as you can see how that particular businesses can continue to adopt that business continuity plan. With the COVID-19 situation, with economic situation, with particular what happened, the factories or the processing, the plant actually shutting down, you can see the impact on the food retailers, because what happened, COVID, supply problem and gonna be demand problem. It's a very clear. So what's happened if high demand and you got low supply, is empty shelf, which is stock out. What happened if low demand and you got high supply? Just before COVID, as you know, food perishable product, you can see with a dairy product, you cannot sell particular milk or fresh milk, more than like 5 or 6 days, say for example, because product is already expired. For bakery, for bread, for instance, banana, say for example, that particular element we need to look at supply chain, food, fresh produce, agribusiness context. And this is particular, we look at different types of methodology we can adopt: econometric modeling, simulation, operation research modeling, statistical modeling. Since I'm actually a mixed method scholar, which means that I can do quantitative and qualitative research. So I think majority in this room, most of you doing quantitative or using design experiment or statistical or simulation. I'm sure you really like numbers or modeling side of that, or food modeling. Since my background, I was graduated in industrial engineering, so pretty much I know very well with operation research and statistical modeling. And the second project is future research going to be about halal food. So food varieties, you can see it depends. Maybe you can see, you know, in terms of the seafood or particular, you know, different types of food products you can look at. And then everybody knows sustainability. Sustainability dealing with the environmental impact in terms of food waste. As you know, food supply chain sustainability, we really want to minimize the food waste. How do we make sure the business can be sustained in order to make sure how do you produce and what you want to sell, make sure really, you know, not having inventory stop. If you have a stop, say for example, it's gonna be food waste because the market is so slow, the market is so quiet, and you got high supply and you cannot sell it actually because the product actually is not really good at all to be consumed. That particularly is become food waste. So that really good research in terms of food supply chain sustainability. The fourth research is about Industry 4. Everybody knows Industry 4. Industry 4 is pretty much about technological advancement. For COVID-19 now, we experienced nearly the last 16 months, but you know, the good thing for Australia, since the last semester, all my students actually having face-to-face. So we only have an online, maybe, you know, for only one semester, which is semester 1 in 2020. You know, so far so good for, we do not have problem, but student can come back to the campus live, and then we still have the online, which is we have a student from interstate or from overseas. They can do pretty much online studying. But this is, we talk about digitalization, supply chain technology, artificial intelligence, sensors, machine learning, robotics. They're all about the supply chain Industry 4. You can see the agribusiness and food sector. Supply chain Industry 4 is a very good topic for research, especially if you are a food science scholar. You'll be able to work with the business scholar, and you'll be able to work with computer science scholar, or from, you know, electrical or maybe manufacturing engineering, because they really more high technological oriented. So that supply chain Industry 4 is a very good topic for future research. And then supply chain analytic, agribusiness and food sector as well. So I think I mentioned before about food waste research. That's one paper already published in 2019 about two ways and not two ways, exploring motivational factors of Generation Z hospitality employed toward food waste in the hospitality industry. This is published in the top tier journal Q1, International Journal of Hospitality Management. All right, so I would like to say thank you very much indeed for listening. So I really want to have any questions or comments, or this is good opportunity for this webinar, even though we have a lot of scholars from overseas, from other countries. You know, this is something very good moment to engage with the scholar. How do we can work together, given we are having you know, different skills, different research capability. Once again, thank you for listening. Have a wonderful webinar. Thank you.
Thank you, Dr. Ferry. It was a nice presentation. Is anyone having any questions? Okay, I think there are no questions. We can move
Yeah,
towards the next
I have one.
presentation.
Sorry.
Yeah.
At least one or two scholars need to ask me question, because I really
Okay.
want to have the webinar need to be engaged. But if no question, maybe I don't know what scholar maybe.
No, Dr. Ferry. Dr. Patricia is having some question.
Sure.
Okay. Yeah, thank you very much for your very interesting and nice talk. I've seen that you have published about trust and milk product, and I'm also researching with among health claim declarations within the related to calcium in functional foods. And I'm not sure if you have worked with the health claims.
It's a very good approach. This is something we can work in the stakeholder research in terms of to have better understanding, right, for the claim, for health claim, because for the trust and commitment, building trust and commitment is a very important, right? It's a building relationship. So this is it's a multidisciplinary research. So what I can see for agribusiness research is pretty much, I know for the food science, you know, it's pretty much is a specific for your research. But if you really want to do, you know, it's a big grant or with the United Nations funding, with a collaborative research, CRC, what we call in Australia, is coming from different discipline. It's what we call multidiscipline. So I think Dr. Pratisha, you're right, because the trust and commitment really to look at the antecedent cooperative behavior variables, like the claims, particular to look at the culture, and then to look at the historical. And of course, when you do that particular research, calcium, for example, how do you look at that particular, you know, element, you know, can be trusted? And that particular, we working together, we need to understand the ingredient element, how what particular stakeholder become trust that you over that product. You know what I mean? Say for example, I'm producer, I'm in the food tech scholar, so I completely don't understand what the consumer, what is the government, whether they trust my product. Talking about vaccine, some other country they got problem with the blood clot, whatever, you know, and this how the particular government or patient, say for example, they trust whether or not that particular vaccine or COVID vaccine, you know, suitable for my body. You know, we need to maybe test, you know, whether significant level 99% or 1% alpha. Of course, we need to grab actually the huge sample, you know, to measure that the trustworthiness, right? That's particular example really, because of course the vaccine rollout for COVID. Now Australia is a very good now for vaccine rollout. So it's now we are in the stage 1B. So we're hoping, we're talking COVID vaccine, you know, in Australia, hopefully by June, July. But all countries doing the same. Of course, we're gonna be, you know, solve the problem, right? So this is something, you know, we really want to work together for all the countries. You know, cannot be blame each other, you know, all the COVID situation, cannot blame this country, cannot blame the government, all those things. But sorry, it's out of context now about COVID, sorry. But this is because still relevant, isn't it? Because I just finished, you know, debate in terms of the education scholar with the COVID, with the vaccine, with the food, you know, scarcity, given I'm in supply chain, because scholar, I really look at end-to-end supply chain, including transport, logistics, manufacturer, food product, from the beginning until the packaging, until consumer eating the product. So we talk about beef pretty much from farm to plate, which means like you got live cattle until you eat spaghetti, right? Until you eat beef curry, for instance. So that is a pretty much what we supply chain, right? So sorry about that, Patricia. So very good question, really, but very good research. I believe if particular moving on for your research, I listened to your presentation. Wow, is a very high level in terms of in terms of your research capability, very expert in your area. So well done, congratulations. So I'm trying to have better understanding. I know calcium is not my area, but very good to understand how, you know, expert your field to share or everybody in this room. So that's one, we are here to share, you know, in terms of knowledge and your research here. Who knows, we can work together. So that's why I put in the chatting box. I share my email address, because I'm coming from Edit Cowan. It's just, few hours different now, travel. So sorry, so I'm talking too much now. Maybe I just give you opportunity for other speakers as well.
Thank you very
Patricia.
much. Thank you for your answer. Thank you.
Thank you, Dr. Perry, for your explanation. Is
welcome.
anyone else having any questions?
Hello, can anyone hear me?
Yeah, Dr. Vinuta.
Yes.
Yeah, hi, Dr. Ferry. It was a nice presentation. I'm from India, so actually I'm doing PhD, so not a doctor basically yet. So my work is actually to identify some like crop improvement methods that I can improve, mainly in sorghum, to improve the fodder quality for feed. Livestock basically. So when you say from farm to plate, do we still go another step ahead, or like before from farm? So like, because what we feed to animals is what we get to eat in terms of meat or milk yield.
Of course.
So, yeah, so how much of importance has been given to that part? Because when I see review of literature for my research, it is like, and particularly for sorghum, it's very scarce. So why is that gap has been developed in this area? Can you please show some light
Very
on
good
that?
question, actually, Vinota. So maybe before to answer your question, hopefully everything's all right in India. So we are so sorry to look at India's situation now with the COVID. So I got a lot of my scholar from India, just look at I know it's so sorry to see in India situation right now. So for COVID situation. So I hope everything's all right you in India right now. Now, in terms of end-to-end supply chain, I completely understand we talk about the animal science. It definitely about animal science. Everything, the ingredient, to fit for cattle, for sheep, or any particular kangaroo, say for example, particularly we really want to look at the finished product, whether, to look at the fat level of the cattle, what is the correlation between what ingredient you just feed them, to look at the fat level, or maybe to look at, quality. So if we look at the quality of that particular producer, right, live cattle, we need to look at what types of ingredient. This is something we look at food science, and food text, and a lot of data need to be captured to tell for the, abattoir or the processing, and to tell the consumer, look, we have a particular product actually, because the ingredient you feed them actually, as a result, actually highly correlated. You can see the statistical significance. So this is particularly, you really need to do a lot of testing and sampling to look at, say for example, you can see Wagyu beef, right? In Japan, for instance, you can see what we compare Wagyu beef and compared to the Australia, maybe compared to Indonesia, with different types of beef, and you can see what types of feedlot. This is something we can work together with feedlot. If you're a feedlot, what types of ingredient you feed them, all your cattle, right? Now, the processing company, they can sell the product and they can see, we can, particular from downstream, from retail, from the market, consumer can tell you, particular the survey with the product in terms of consumer behavior, the market analysis in terms of the product actually performance, and then back to the what we call the agriculture conference or congress or research and development, working together to capture that data. It is not 1 month project. It is not 6 months project. So this is something you really need to have a different stages. But I just want to give you like, the process really. Because my expertise in the business, agribusiness, well, pretty much if I've developed working paper together, I'm strongly contributing in the methodology. I'm working with the food science from the other food, school of science. Because some of them, they don't know about modeling, because I know about operation research simulation, statistical modeling. as long as they can give me this to the objective function or identification problem, what variables in there. Once I understand, then I can develop the modeling. Then I can look at what is the results. I can use Mathematica, I can use MATLAB, I can use Stata, I can use Minitab. Different types of software can do that. This is something my contribution to the particular research, because I'm actually good in the methodology part, but I'm not good in the food technology, food science. Like Patricia, you expert in particular this, and then you maybe do not know particular one element of methodology. We can, you know, try to come up with the particular results. Very simple when we do research, collecting, analyzing, right? And then pretty much, well, this is pretty much we are doing PhD, we're doing research. Even though I never finished for my study, I like studying. I really want to do my postdoc because I'm now completing nearly 4 different postdocs. One about food supply chain, one about food sustainability, one about food waste, and the last probably project really dealing with the food COVID impacting on the, you know, supply chain in the food industry, food and beverages. You can see now with the COVID, because, you know, with the food safety, food security, very, very important.
Yeah.
Okay? Yes, sorry, I just know I don't want to share all over the place, but yeah, in terms of technicality, of course, cannot just answer your really 100% your answer, but I just want to give you the process.
Yeah, thank you.
You're welcome, Finn.
Thank you, Dr. Ferry. Is anyone else having any questions?
Very good, Jessica. Since yesterday I looked at the webinar, very good. I can see, even though in difficult situation, so good to have this conference. Actually,
Thank you very much, Dr. Ferry.
Yes, we need to promote more engagement, promote more papers, promote more research, right?
Yeah.
To work together with government, to work together with FAO, you know, United Nations Organization. Now, because what happened, they just run out the people having good skill in the area of food, right? So this is very
definitely we will be providing platforms, and we will be organizing webinars on food research.
Yes.
So we will move towards the next presentation, and it is from Dr. Marko Vinskovic, University of Zagreb, Croatia.
Yes.
Hello, Dr. Marko.
Hello.
Are you ready for the presentation?
Yes, I'm ready. Study and go.
Yeah, so I'm making you the presenter.
Okay.
And you can share your screen and start your presenter.
Okay.
Yeah, it's done.
Okay, wait a second. I have to find first. Okay, wait.
Okay.
Now I could share it. Okay. And now I will start with my diagram projection.
Yeah, it's visible now.
Okay, so hello, my name is Marko Vincekovic, and I'm working at the Faculty of Agriculture, which is a part of University of Zagreb, and I will have a talk about production of functional food by application of microparticles filled with bioactive components during the agricultural production. So first of all, I would like to start with the second. I need to wait. Okay. I don't know how to. Wait. I don't know how to start to move to move further, because I can't.
Hello,
Jessica.
Is there any? Yeah.
Yes, because I can't move my slides further with my computer, you know.
Okay, you can do one thing. You can send me a presentation. I will play the slide.
Wait a second.
And
maybe I have these options or not. Make a presenter into.
You can click on the mouse, like it will move forward.
Okay. Oh,
Scroll the mouse.
Okay. It is going now. Okay.
Okay.
So hello everybody. My name is Marko Vincekovic, and I'm a part of University of Zagreb, and I'm working at the Faculty of Agriculture. So my talk is about the production of functional food by application of microparticles filled with bioactive components during the agricultural production. So first of all, I would like to introduce my faculty. She's a part of University of Zagreb. We were founded in 1919 and we became independent on 1959. So we have altogether 423 employees and also 2,619 students on different level, from undergraduate, graduate, and postgraduate level. So what is very important for the Faculty of Agriculture, we own 6 experimental stations, which are used for scientific, technical activities, research, and practical work for teachers and students. And altogether, we have got 28 departments. So you could see that we are a very interdisciplinary institution. So I will start with introduction. So what is the encapsulation? So the encapsulation is a packaging technology of solid, liquid, or gaseous substances into small particles, which could be spheres or capsules. And they have got a different diameters, from several nanometers to several millimeters, and they have got a content which is released over a certain period of time, and of course under the certain conditions. Okay. So why encapsulation in agriculture? Because encapsulation presents an innovative approach to stimulate the production of secondary plant metabolites. Also, this is a new way to fortify plant defense against different types of pathogens and pests. And also, with encapsulation, we could protect the bioactive components which you could find inside these microparticles. So what was our research goals? So our research goals were the preparation of microparticles, so microspheres or microcapsules, which are loaded with biological or chemical agents. We optimized the preparation of these formulations, and also we applied these formulations into different types of agricultural systems. So we increase the defense mechanism, and also we produce the functional food. So what was our materials and methods? So we are using ionic gelation methods in the process of production of microparticles, and also we are using polyelectrolyte complexation. So we could encapsulate different types of bioactive components into alginate microparticles or other biopolymer carrier. So it could so we could encapsulate chemical agents, microorganisms, essential oils, lipids, biomolecules, and single compounds. So first we encapsulate biological agent, which is Trichoderma viride, and Trichoderma viride is very important because it is a beneficial fungi, and it is also a plant growth promoter. It is a very aggressive microorganism, which is very aggressive to other pathogens. So at the same time, it is a plant growth promoter, but also it protects the plant from other pathogens. So this is a process of production of microparticles. So as I said, it is a two-step process. So first we have got this process of ionic gelation, and after that we have got this complexation process with second biopolymer. In this case, it was a chitosan. So we had several variables. So we have got different particle sizes from 160 to 2000 micrometers. So we have got a different concentration of gelling cation solution, and also we have got a presence of a biological agent, which is Trichoderma viride spores. And also we add this second layer of chitosan coat. So this is a sham at which you could see that first we made different types of microspheres, then we make investigation about their interactions, then we make this process of release behavior, and also we measured different types of physical chemical properties. So we have got very interesting results. First, we investigate interactions between Trichoderma viride spores and gelling cations, and we find out that these Trichoderma viride spores are negatively charged. So if you increase the concentration of calcium ion, which is a positive charge, then you could make this decreasing of the negative charge of Trichoderma viride spores, and they start to make different types of aggregates. So with this increasing of the calcium ion, we did not reduce the activity of Trichoderma viride. So we measured the interactions between biopolymers and encapsulants, and we made that by Fourier transformer infrared spectroscopy. And we find out that the main interactions between bioactive components and biopolymers are mainly by electrostatic interactions and by hydrogen bonding. Also, we measured in vivo release of active compounds, and in both cases of microspheres and microcapsules, we have got a diffusive mechanism of releasing of bioactive components. With higher concentration of gelling cations, we increase the releasing, but with second layer of chitosan, we show decrease the releasing of bioactive components. Also, we made a completely physical-chemical characterization of microparticles. So we use several microscopes like scanning electron microscopy, confocal microscopy, and also we used optical microscopy. Also, you could see that that these Trichoderma viride spores completely survived this process of encapsulation, but also they start to spread and to grow inside these microparticles. Also, in the case of microcapsules, you could see here on this third picture that we have got this second layer of chitosan on the microsphere, which is built by sodium alginate. Also, we made this investigation of the surface of the microparticles with atomic force microscopy, and we find out that that we have got a granular surface structure. And we could also see that this microsphere surface roughness is smaller than microcapsule surface roughness. After that, when we made all this investigation, all this optimization, we start with the application of the optimal microparticles on different plant cultures in different agricultural system. So you could see how we apply it. So first, we apply these different types of formulations on the green lattice, and so we use different types of formulations, and we apply it for 2 years in the open field and 1 year in hydroponics. So this article is already published, and it was published in the scientific reports under Nature. And we find out that with this application of microparticles, we increase the level of the secondary plant metabolites. In this case, it is a chlorophyll. And we increase the level of chlorophyll in some cases for almost 86%. Also, we increase the level of polyphenols, and also we increase the level of antioxidant activity. Also, we apply different types of microparticles in the process of cultivating of grape vine. So in this case, we use microparticles which contain calcium, magnesium, and Trichoderma viride spores. And again, we increase the level of the carotenoids and chlorophylls in the Vitis vinifera leaves. And we find out that we increase the level of chlorophylls for the almost 86%, just like in the case of the lettuce. Also, we increase the level of polyphenols and antioxidant activity. Also, we apply different types of microparticles in the process of production of strawberries. This is in the so this was a hydroponic way of growing of strawberries, and again we increase the level of polyphenols, flavonoids, and antioxidant activity. Also, we increase the total content of anthocyans and flavonotriols. Also, we apply different types of microparticles on tomatoes. So we use several types of cultivars for 2 years, and we apply it in the different agricultural systems like conventional and hydroponics. And we again have a very good results because we increase the level of very important secondary plant metabolite, which is a lycopene. So we increase the level of lycopene in some cases for almost 200%. What is our ongoing research in my laboratory? So now we will apply microparticles on strawberries, and these microparticles will contain zinc and iron, and they will be applied foliarly in hydroponic way of growing of strawberries. Also, we will apply microparticles which contain calcium and gibberellic acid and indole acetic acid during the production of Cape gooseberry. And these plants will be cultivated in open field. Also, what have been done in the last 2 years, it is the application of microparticles which are filled with copper ions in the case of grapevine protection from peronospora. You know, we have a very good result because in this case, when you are going in the process of protection of grapevine against peronospora, then you don't need to think if it will be a rainy day tomorrow, because these microparticles will be attached on the leaf and it will not be so easily washed. And also, if we shall have a rainy day, it will be even better because then this copper ion will go out from these microparticles, and of course the grapevine will be protected. So what is the final remarks? So we produce different types of microparticles which are filled with chemical and biological agent. We made completely physical chemical characterization. So we find out that there is a complex interactions between biological agents and chemical agents. Also, we find out that we have a diffusing mechanism of releasing of bioactive components from microparticles. And also we find out that we produce this new microparticles formulation, which has got a 3-in-1 effect. So at the same time, it is plant protection, plant nutrition, and also it is a time release mechanism. And it is, of course, an agroecological product because it is completely biodegradable. And also what is very important, these formulations were produced by a green chemistry process. So with these applications, we could increase the amount of certain bioactive components in plants, so we could produce the functional food. But also, what is very important, we could protect nature and groundwater from other organic chemicals which are used in agricultural production. So what are the benefits of this technology? So we could protect the bioactive components. We could increase the easier uptake of these components. We could stimulate the secondary plant metabolite synthesis. We could protect the plant from other pathogens. And with this encapsulated bioactive components and with this application of microparticles, we have got this new innovative approach in functional food production. At the end, I would like to say some awards which we get in Croatia and also in Germany. So we got this award on Zagreb Startup Factory, which was held in 2018. Also, we got this award from Zagrebačka Banka, and also from the Climate Kick, which so we have been the first team from Croatia which was a part of this Climate Kick accelerator. So at the end, I'm sharing some beautiful pictures of my country. And if you have time and if you have a goodwill, you could visit my country. So thank you very much for your kind attention, and I'm waiting for your questions. Thank you.
Thank you, Dr. Marco. It was a good presentation. Is anyone having any questions? Okay, so we can move towards the next presentation.
Thank you.
It is from, thank you, Dr. Marco. And the next presentation is from Dr. Demon Gisbrick from University of Colorado, Canada. Hello, Dr. Demon. Are you ready to give the presentation?
Yeah, absolutely. First, I want to full disclosure, I am not a doctor.
Okay, sorry, yeah. Mr. Demon.
No, not a doctor. I do have 20 years botanical experience as a both medical patient and grower with the cannabis plant.
Okay.
So I guess, yeah, this is a great spot to get into my presentation and my keynote. So if I can share the screen.
So I make you a presenter. You can share your screen and start your presentation.
Okay.
Yeah, it's visible.
Can you see my screen?
Yeah, it's visible.
How do I minimize all of this? Okay. Oh, perfect. There we go. Okay. Everybody can see it?
It's visible.
Okay. So here we go. As I mentioned, I am not a doctor. And I guess we'll just start with the introduction here. The agenda today that I'm going to go through is I'll explain who I am, what I've done, and where I'm at with things, how I figured out the efficacy, dosage, and all of that surrounds cannabis, some of the methods that I use, examples, and a conclusion. So, as I mentioned, again, I'm not a doctor. In 2001, I was injured in a pretty serious motorcycle accident. Throughout that process, I was diagnosed with chronic pain and post-traumatic stress disorder. In Canada, we did have a medical marijuana program. However, the doctors had no idea about efficacy, any of the knowledge, safety, things like that. So essentially, there was no formal way for me to take this approach. I had to learn all of this on my own, and within Canada and even internationally, there was very few places that you could go and learn about the plant. So when legalization happened in Canada, I was able to start working for a company in Canada that educated a lot of the people getting into the industry and sharing what I know. So I have consulted on hemp farms in my area, 4 plant grows, which is for the legal home grower in Canada, all the way up to facilities that have 40,000 plants in them. I do educational services to licensed retailers and cannabis companies throughout the world. The highlight to my career so far is I've consulted to a few First Nation cannabis organizations, their stores, and then, you know, the people that run the regulatory boards that oversee it. Currently, my new project that I'm working on is proteomics and epigenetic signaling within the cannabis species of plants, and that's something relatively new to me. There's a lot of methods and stuff that growers will use that are stressful to the plant. So with the epigenetic signaling, we're able to eliminate those stressful processes and kind of trick the plant in thinking that we're stressing it. So 20 years ago, when I got ran over and after my doctor said, you know, I can't sign any sort of prescription or anything, I have no idea about the efficacy, safety, dosage, and the quality. So the things that I figured out that I had to know when I was looking at a particular inflorescence and whether or not it was what its use was for was within cannabis, there's generally daytime flowers and nighttime flowers that you can use. The dosage, that's something that really up until about 2-3 years ago, a lot of people had no idea what they were taking in. If you go back to the pre-prohibition and pre-ibuprofen days, the cannabis was quite, it was frequent in a lot of medications, and the way that it was done then was pretty much just a titration of a dose. Safety within cannabis, there are no recorded overdoses leading to fatality. Adverse reactions are plenty, but nothing that's really requiring hospital. Last few years, there's been incidences of cannabis hyperemesis syndrome versus cyclical vomiting syndrome. There's a bit of anecdotal evidence surrounding that it's actually not the cannabis itself, but it's rather an input that's put in. The good thing about living in Canada is a lot of the inputs for cannabis have to be regulated and come with a registration certificate from the Canadian Food Inspection Agency. So within quality, especially in Canada, there's 2 ideas of thought. There's the production, your standard operating procedures, and then what the consumer defines as quality. And it's a drastic difference. There's a lot of cannabis within our legal medical system that it checks all the boxes on the production quality, but the end user they would not deem it quality. So nomenclature and taxonomy with cannabis is highly debated. There's 2 schools of thought behind it. One is the Linnaeus-Lamarck traditional way of doing it, and the newer one is by Clark and Merlin, and it's narrow leaf drug all the way up to broadleaf drug, and it's the visual characterizations of the plant. So going back to how I figured this out, there are great limitations of being self-taught. I've had to figure out a lot of the correct botanical terms, Sometimes I use the wrong word. After 20 years, I like to think I'm getting there, and I'm not doing that anymore. The information at the time and even now, some of it is very reliable, and some of it is false. And then within a lot of the cannabis science that's going on, some of it is irrelevant. So going back, there was a lot of trial and error on my end that I had to go through to deal with some adverse reactions. And generally what that means is you're using the wrong flower at the wrong time. And then now there's a lot of non-accredited resources for learning how to, dictate quality, grow cannabis. And the accredited is starting to pick up, which is really nice. So some of my methods, generally I use a classic wet chemistry approach, natural plant medicine. I look at it as such like that, less of a pharmaceutical and more that this is in line with grapes, cucumbers, things like that. There are industrial wet chemistry approaches to satisfy pharmaceutical and the forensic approach. You can use UV microscopes, doing a microbiological analysis. And then the latest technology that's come along is near infrared spectra analyzing, which has been quite great to use. And there are limitations within the traditional forensic methods, thin layer chromatography, gas chromatography, high pressure liquid, or high performance, sorry. And those limitations are like with NIRS, it's the device and the machine that's looking at the output and giving the data, whereas with the traditional methods, there's still a human room for human error. So within the qualitative data, there's a lot that you can see in the plant that will determine its use. Ethan Russo and a few other people, they've put a lot of time and energy into the research behind your olfactory system, and where you're smelling the plant can help you dictate when you're supposed to use it. The other thing is within the medical cannabis sector, because we've been so limited for the last 20 years, it's really the patient that ends up deciding if they like that. Your nose will know. Okay, so visual characteristics determine the efficacy and the quality. If it looks horrible, it's going to be an extremely horrible flower to use. So within the inflorescence, the characteristics that we're looking for, the pistils, any contamination, presence of any flavonoids that you can see like carotene and anthocyan. And then the stage 2 drying phase, like how does the flower feel, and residual ash that's left over. So the classic wet chemistry approach within the plant, you're looking for the pests, the contaminants, and then you can use either form of taxonomy. Both do apply. Clark is more accurate due to the amount of polyhybrids. But if you are looking at land races, so that's where, that plant is feral in that area and it's been growing that way for quite some time, it's a lot easier to work with land races determining the efficacy and the quality. So image alteration, as you can see here in the example, it draws out those anthocyans, it lets you see the flavonoids, and it will also draw out possible contamination. None of these examples really have any signs of issues, and you can see based on their leaf structure what they're used for. These top ones are more of a narrow leaf drug type. This is a broad leaf drug type. Now we're in between a broad and a narrow leaf drug type there. So that would, and it kind of leans towards how to determine your use. So with microscope or using a microscope, it's quite easy to see the pests. Here you can see a pest actually dead inside the flower, and the trichomes appear to be sprayed with something. You're also able to see flaws in the flower, the viruses and the viroids, things like that. You're also able to see the high quality flowers. We have found that when the trichome gland is a milky color to here, the amber ones tend to be higher in CBN, but this would be, you know, a pretty potent THC trichome head. So the interesting stuff and where all the data comes from that I was able to take 20 years of independent research and tie it all together is using near-infrared spectroscopy. Company I work with and have partnership with is called Gemacert. They're based out of Israel, great company, extremely good data bank. So here we have a flower that is considered a Cannabis indica afghanica polyhybrid. This would be under Clark and Merlin, it would be a medium leaf drug type, daytime use. Takes about 14 weeks to flower off during its flower photo period. But the interesting thing is with this flower is regardless of its potency, you see, we have generally it's THC based and there is no CBD located in the flower. But regardless of where that flower came from on the plant, the use is always going to be the same. Really, the only thing that's going to change is your dosage. So again, we have a landrace C indica here. And you can see, again, it's all over the place. But this flower, it would be a nighttime use flower because the leaves are broad, there's few crystals. It's high in anthocyanins, so we know there's research out there that dictates that anthocyan can be used in chronic pain situations. And then here we have a Cannabis indica afghanica. It's about a 10-week flower period. Very little CBD located in this flower, but again, the THC levels, all that really comes into play with the lower of a THC level is you need a higher dosage, but it's a very marginal higher dosage that you need. Again, here's the data with the flowers all overlaying over top. So now we'll just say, for example, this one here was contaminated flower, had aspergillus in it, and this flower here was clean. The flower that has aspergillus in it is going to have a lower efficacy because it's contaminated. The user isn't going to enjoy it and they're going to end up with an adverse reaction, whether that's a twisting in the chest sensation, coughing fit, or anything like that. However, the lower cannabinoid ratio flower is going to have a higher efficacy simply because it's cleaner and the user is able to ingest more of it, whether they're inhaling it or via means of inhalation. So just a little bit of an example of how I go through my process. The first information that I always gather is where it was growing, who the grower was, the strain name assigned by the farmer, just as a way to identify, you know, details. Its harvest date, its package date, you know, so that we can determine how its window of freshness. What was present in the flower for aroma. There's a lot of research out there, especially by Ethan Russo and others, relating to the entourage effect and how terpenes kind of dictate use on the flower location. I tend to prefer to look at visual characteristics alone to dictate that. And genetic failures that are present. Flowers that have anthers and have turned atavistic or monoecious, you want to stay away from them. They're going to have seeds in it. The seeds, cannabis seed oil is carcinogenic when it's combusted. So these are things that you really want to avoid because it will bring down that efficacy, it brings down the quality, and it greatly affects the safety of the flower. So the health conditions in the flower, the pistil health, the trichome health, these trichomes are all unhealthy. Some of them, because of that milky color to it, they do look like they've been sprayed or had some foliar application done to them. We're looking for visible microbiology. Botrytis is very common within cannabis, powdery mildew. Aspergillus is another common one that ends up happening. Evidence of pests, evidences of pesticide use, you know, so we can't, visually we can't get down to the ppm, but we're able to deduce that, you know, there are pesticides being applied just based on biological controls and indicators. You know, if you're seeing something like this in this top right side there, and then you turn around and you find, oh, here's a bug, generally I'll go back to the grower and I'll question them on it. And what will happen is the grower will end up looking at the floor and admit, yeah, I sprayed this, I had an outbreak of spider mites or thrips or hemp borer beetle. Color uniformity is something else that you want to go for, and that's, you know, like, it's just like buying grapes, bananas in the store, strawberries, things like that. It's quite easy to do visually. And then presence of foreign material, that's something else that we see a lot of, whether it's a strand or fiber from, you know, an employee's uniform working in the facility, all the way down to the home grower that ends up with dog hair and things like that. So the glandular health, the stalk health, the color of the gland, these are all things that are extremely important when you're dictating and looking for efficacy, quality, and safety. You want healthy flowers, you know, that are formed properly. Ornamental use, PGRs, things like that. Synthetic auxins, those are things that kind of find their way into the market. But, you know, if you're looking for a naturally formed inflorescence, you're not going to have that problem. Cannabis, as some of you may know, is extremely good at pulling heavy metals out of the ground. They store the heavy metals within the plant. So there's two processes that are required to pull those nutrients out. One is called chelation and the other one is flush. Chelation pulls the heavy metals out of the plant and the flushing pushes it out of your pot or your bed or away from the flower so that you end up with a clean flower free of aesthetic and heavy metals. Hand trimmed flowers are always higher quality. In the leaf material here, you'll find higher doses of chlorophyll, and especially if the grower didn't cure it properly and draw that chlorophyll out with the ethylene. And then again, I've already touched on this proper inflorescence structure. There's a lot of, like in Canada, we're good because Canadian Food Inspection Agency does regulate and oversee the program, but you will see in other jurisdictions where there are ornamental fertilizers being used on the flower and they really shouldn't be. So covering off on strain names, I'm going to give you guys an example here. These two flowers have exactly the same strain name. They're polyhybrid F2 generations. The THC and the CBD levels across the two were fairly consistent and similar. The terpene profile, where the flowers smells, that's very similar, but the effects vary. They're on opposite ends of the spectrum. This top flower here is more of a daytime use flower. And the bottom flower here is more sedative and relaxing. So why is that? Well, within the F2 generation, we don't have uniformity yet in the genetics. So we have, you know, a copy of the mom or from the P1 generation and a copy of the dad from the P1 generation. And what happens here is the plant structure is really what you want to look at. You know, avoid the smell and everything. because you can just, myself, and you can just tell this one leans more towards narrow leaf drug or sativa. And the bottom one here, because of the fewer pistils, it leans towards broadleaf drug or cannabis indica. The other thing is both flowers, you can see the presence of anthocyan in it. And again, anthocyan, there is research out there saying that it's great for chronic pain use. Myself, what I've been able to determine here is that the flower on the top works great for daytime, flower on the bottom, great for nighttime. So, and again, back to one's Afghanica dominant and one is indica dominant. So the wrap up. In terms of efficacy, what I've found over the last 20 years is strain names don't really provide an accurate representation of effects and appropriate applications for a particular medical condition. Throughout my time is looking at other people's cannabis in the last 2-3 years, many growers were using bacterial supplements improperly as foliar sprays, resulting in cannabis with a higher microbiological count. In one instance, the lower the potency that was clean had greater efficacy than higher potency samples that had contamination. Higher potency cannabis that was clean had an even greater efficacy than samples that were contaminated. Knowing flower photo period for a particular inflorescence allows for proper medical use. And I'll come back to this example here. This flower down here was finished in 8 weeks, and this one took about 10. So like that flower photo period makes it, you know, you can tell the ones that take longer to flower, they're more of a daytime use flower. So the other thing I wanted to touch on is within cannabis, there is no universal standard on quality. There's no pharmacopoeia that's accepted universally, and there's no monograph. Aside from Canada and a few other jurisdictions in the world, it's a completely self-regulated industry. And again, it's changing, but a lot of the information that's out there is anecdotal, and that is where I come in my limitations of not being formally trained, but I am working with a lot of great people that are helping me take my anecdotal evidence and we're turning it into real world evidence. So if there's any questions, oh yes, and resources. I get asked a lot what books people should get and what they should read. Pretty much the backbone of cannabis botany is by Robert Connell Clark. It's Marijuana Botany, and it's an advanced study. It was written in 1981. Not much has changed. The nomenclature is still really debated. The Hashish Eater by Fitzhugh Ludlow. It was originally written in about the 1800s, around the same time as The Great Gatsby. The book is kind of it does cover a lot of dosage of hashish. The Pot Book by Julie Holland. This is another great book for getting into the medical uses of cannabis. One of my most favorites, Cannabis Evolution and Ethnobotany by Robert Clark and Mark D. Merlin. This really opens the doors to the narrow leaf drug, to the broadleaf drug idea of taxonomy, and it works. The American Herbal Pharmacopoeia on Cannabis Inflorescences. I enjoy this particular monograph and pharmacopoeia simply because it simplifies how to grow cannabis and doesn't really get too overly complicated like the UN Drug Pharmacopoeia or the European Union Drug Pharmacopoeia. And then Ethan Russo's study on taming THC and the entourage effect, that was a really great study. And then one of the books that's recently come out, and it's from the Trichome Institute, and it's called Interpening. Interpening is that process of using olfactory senses and things like that, but within a cannabis sommelier perspective. So that is everything. If anybody has any questions, I'll greatly answer those. I look forward.
Thank you, Mr. Damon.
Pardon me?
Thank you, Mr. Damon, for your presentation.
Yes, then thank you for.
It was really informative about the cannabis. So is anyone having any questions?
Yeah, hi, Vinita here. Hi, Damon.
Yeah, Dr. Vinita.
Yeah, hi, Damon. So I am not sure I understand the whole thing, but that's a really lengthy, very lengthy period that you spent over researching over marijuana. So my question might be naive, but what do you think there is a difference between, if there is a difference between consumption of leaf, flower, or seeds, if there is any, in terms of medical purpose? Why is your research is mostly focused on flowering behaviors and flowering patterns and flowers for medicinal purpose?
That's a good question. So what happened when I was talking to my doctor, he, and this is going back to 2001 when I was trying to get the Health Canada authorization from him. He put his pen down and he put his hands up and he's like, I don't know anything about this, right? So a lot of what I focused on was clean flowers, right, while I was growing in that. And it really wasn't up until when cannabis was legalized in Canada where I was seeing all these other growers and the contamination in their flowers, and why myself as a medical patient, I wasn't enjoying it. So we looked at it, and it was, you know, there was things like covered in mold, spider mites, foreign material, all these things that just kind of, as a medical patient, it wasn't a clean flower, so it wasn't enjoyable. The market, even the regulated market right now, is extremely problematic. There's mislabeling issues. We're finding that, you know, when you buy something and it's got an HPLC label on it of 23% THC, really the range of phytocannabinoids within that bottle are 13% THC to maybe 19% THC max. So it's not a naive question. It's because I'm not formally trained yet. Like, I'm going through the process, and there are great companies and institutions that I'm working with that are helping me get that formal training. But because I was able to take a different approach to it due to the lack of my formal training, I was able to figure out, you know, this is a natural plant medicine. It grows wild in many areas of the world. There's still, you know, indigenous cultures that are using cannabis as a natural plant medicine. And I wanted to focus more on that sort of way, like, of quality, rather than, you know, a actual quality assurance by definition that way. Does that answer your question at all, doctor?
Yeah, thank you. And yeah, I'm neither a doctor, so still a student. Thank you for that.
Okay, thank you.
Thank you, Mr. Damon. It was an okay. So is there anyone having any questions? Okay, thank you once again. So we will move forward for the next presentation from Ms. Vinuta K. Somgowda, ICRISAT from India. Hello, Ms. Vinuta. Are you ready for the presentation?
Hi, yeah, Jessica, I'm ready.
Yeah, I'm making you the presenter. Yeah, it's done. You can start your presentation by sharing your screen.
Yeah.
Yeah, it's visible.
Is it visible now, Jessica?
Yeah, it's visible. It's visible.
Hi, hi everyone. Good afternoon. My name is Vinuta. I'm a PhD student. I'm working in ICRISAT. It is International Crop Research Institute for Semi-Arids and Tropics. The headquarters is based in India. So I am working on my PhD. So my PhD objective is to identify candidate genes for fodder quality traits in sorghum bicolor. So as sorghum has an innate drought resistance nature, considering its origin from Africa. The crop is mainly cultivated in Asia and African continents. This is mainly for food purpose, that the consumption of grain is high in these countries, continents, while in few other developed nations, we see that there is a popularity gained by sorghum for its second generation biofuel production. However, in other countries where in the semi-arids and tropics, not only the crop is used as food, but the crop residues is also used as feed for livestock. So the crop residues that are used as feed is actually low in quality in terms of mainly digestibility of the feed. And this is highly affecting the yields, maybe milk or meat. So this affects the farmers' income generation because they depend in semi-arids and tropics in particular, because they depend highly on additional income source by depending on livestock. So it's very important for us that we try to help them by improving the feed for the animals, livestock. It's also reported by Blumel that even a 5% variability of digestibility has an effect of price premium changes up to 20%. And so we thought it is really necessary for us to work on this. So I have picked up this as my PhD topic. There are several dual purpose sorghum crops that has been coming up, but still as a grain is a main component in these countries, focusing on quality I think is more important at this point of time. There is not much research done on this one, particularly in sorghum. I think apart from 2018, the one that is reported by Lee et al., where they have focused on sorghum, and they have worked on photo quality, reporting 5 traits that is important to determine the quality. So I'll be touching upon those as well. The current materials and methods that I have used is I have used a recombinant inbred line population that we identified for contrasting parents. That was a preliminary study before this, in which we determined identified 2 contrasting parents, and we developed a real population. So this real population consists of 330 entries, including parents and a few local checks. The experiment was conducted for 2 years in an alpha lattice design with 3 replications each under control and stress. Oh, sorry. Another major issue is drought affects the fodder quality. Less like drought usually reduces the quality of the fodder, not only the quantity but also the quality. So, and sorghum is very commonly encounters with terminal drought stress. So we induced here, we made the terminal drought stress by cultivating the crop in post-rainy season, which because the soil moisture is runoffs and the sorghum has to suffer terminal drought stress. So we took up the cultivation experiment in post-rainy season, and we still had two options to cultivate the crop in normal conditions by providing water, and the other one is to provide stress at booting stage. That's the only difference between the control and the stress plots. Otherwise, everything was similar. And once the maturity is reached, we harvested the biomass. It was dried and then grounded finely, and then we used near infrared spectroscopy to scan these biomass samples. This is a destructive method that has been optimized and identified by International Livestock Research Institute. The headquarters is in Ethiopia, but has a hub in here in India as well. So we worked in collaboration with them and performed this phenotyping. So once the phenotyping was done, the genotyping of the real population mentioned earlier was performed. using the DNA extraction was performed using CTAB method, and GBS sequence was performed, and we had used SNP markers. For the raw file, we have around 290,000 sites. Then we filtered it around 90% coverage of site based on sites, and we have 1,141 SNPs. So these 1,141 SNPs were used to generate linkage maps, and further down the line QTL maps were generated using ICM software, using COSAMBE function. That's the function we use for QTL map generation. And any significant QTL above 3 LOD was used further. Then we, using this significant positions of QTLs, we generated, we scanned or blasted these details on phytosome, where we have sorghum reference genome available to identify if the gene has related to any important pathway or gene that could really help us understand the behavior of these fodder qualities. Once this was done, maize is another interesting crop, because that is mostly used as fodder quality, the top crop used as fodder feeding in these areas. But the main constraint is that it is highly demanding in terms of nutrient and water. So we and there are few research that has reported that sorghum performs on par in terms of feed quality to maize. So we tried to compare maize and sorghum here, and we have obtained few interesting results. I think that could be useful for us to develop further this further. So yeah, so if as you can see here, the results I have shown. Okay, I have for want of time and space, I have not included the statistical results, but I'll go ahead and say that the correlation, Pearson's correlation was performed across these traits. There are several traits that we recorded, but these two key traits, one is the quantity in terms of dry weight, and the other one is the quality in terms of digestibility, are the two things that I'm reporting here. So we found from the Pearson's correlation that these two traits are not positively, neither negatively related to each other. So that means that there is no trade-off, so we can still go ahead and increase the genetic gains of these traits by not losing any of them. So, and with the QTL mapping, as I said, performed with the help of ICM software. If you can see here, under control conditions, dry weight has provided 3 important QTLs, and all of these reported found QTLs is positioned on chromosome number 7, and similarly under stress for the dry weight it is under 7. However, for IVOMD it is on chromosome number 1 and 8. If you can see, I have provided here the QTL maps. Here, this is the chromosome number 7. I'm sorry for the image is very blurred, but this is the chromosome number 7, and if you can see here, this is the hotspot. This is the hotspot that has been observed in our current study, and when based on the review of literature, I have seen the chromosome number 7 has been reported even earlier for identification of dry weight related genes, that is either direct to plant height or flowering or anything that controls dry weight at the end of the lane. So we were very seen very good results in terms of this one. And here we have found IVOMD in chromosome number 1, and in chromosome number 8 also we have found some IVOMD genes. So this was one of the good results that we got. In compared to sorghum, if you can see here, all the chromosomes has one or the other genes for these two traits that has been found synteny in the maize. However, chromosome number 5 has no sharing in these two traits with respect to maize. But however, rest of the genes has some synteny or some ortholos found in maize as well. So the conclusion is that if we try concurrent improvement of both yield and quality in sorghum is possible. The genomic regions, whatever we have identified and annotated, can further be used in marker-assisted breeding to improve the crop improvement. The synteny between maize and sorghum for fodder quality shows developmental and evolutionarily conserved genes between the crops. So further, this can be utilized to increase the pace of achieving the crop improvement for breeding. Yep, I am done with this, and thank you. If there are any questions, I'd love to answer.
Thank you, Ms. Vinuta. So is there any questions? Okay, I think there are no questions from anyone. We can move forward for the next presentation from Dr. Leonie Dreiss from Hochschule University, Germany. Hello, Dr. Leonie.
Hello, thank you. And I'm not a doctor yet, but thanks.
Yeah, sorry, Miss Leonie. So, are you ready with your presentation? Okay, I just make you a presenter. You can share your screen and start your presentation.
Okay.
Yeah, it's visible.
Okay, great.
Can you zoom in? Like it's
yes,
not visible, your poster.
Yes, I will zoom in. So, thank you for giving me the opportunity today to show you my poster. So no worries, I will zoom in a second, but I want to show you the first poster at first. So, better now? Okay.
Yeah, it's perfect.
Okay, great. So, since August 2019, I'm a PhD student in the Department of Soil Science and Plant Nutrition at the Hochschule Geisenheim University in Germany. And in my project, I study the microbial diversity in the rhizosphere of grapevines under different experimental setups. And I don't know how much you already know about the rhizosphere. You can see here. Of grapevines or in general. So I would just like to touch on that again very briefly. So you don't have to read all the background story. I just will tell you the background story for the poster. So first of all, the rhizosphere was already described quite well in 1904 by a German agricultural scientist, Lorenz Hildner. The rhizosphere is the soil volume densely colonized by microorganisms, which surrounds the roots of legumes. And according to today's definition, the rhizosphere is the part of the soil that is physically, chemically, and biologically influenced by the living root. So in general, this means an area up to 4 millimeters around the plant's root. And this area is of particular interest because it is here that the interaction between the soil and the plant and the microorganisms takes place. And for sure, you can find many different organisms in the rhizosphere. On the one hand, of course, bacteria and fungi, as well as yeast, and also some archaea and Euromycetes, and you will find protozoa or eukaryotic one-to-few cell organisms, which include the protists, for example, but you will also find nematodes and microarthropods such as springtails or mice. And of course, you will also find various algae. So you see within the soil anyway, but even more so within the rhizosphere, there's an incredibly high genetic diversity. And there are many positive interactions in the rhizosphere between the microorganisms and the plant, for example, when it comes to nutrient uptake. So some microorganisms also make certain substances available to plants in the first place. A particularly good example of this concept are mycorrhizal fungi. And these fungi form a symbiotic relationship with plant roots and supply the plant with nutrients such as phosphorus or nitrogen, and improve also the water uptake. And in return, they receive from the plant assimilates from the photosynthesis that they cannot produce by themselves. And they can also help the plant defend itself against pathogens. And in addition, the rhizosphere microbiome can make the plant more stress tolerant. These microorganisms also have to come from somewhere, and that happens through something that is called chemical crosstalk. So the microorganisms know they are needed, or the plant sends signals to attract certain microorganisms, and this colonization is called rhizosphere competence. So, and for the purpose of studying the microbiome of the rhizosphere of different grapevine rootstocks, we did this study design. And So we were allowed to sample an experimental vineyard, which you can see here on the left. So the grapevines are planted in a soil mixture in cylindrical tubes with a diameter of 15 centimeters and a height of 120 centimeters in the year 2015. You can see them here on the right. And usually grapevines are grafted onto different rootstocks. And the special thing about this experimental vineyard is that these rootstocks are not grafted, so they don't have a scion like grape varieties like Riesling or Cabernet or whatever on them. So these are just the rootstocks. And we characterized, or we used 4 different grapevine rootstocks, 4 different genotypes, which are commonly used as grapevine rootstocks. And we took 1,103 Paulson, 140 Ruggieri, 169 49 Coderc, and Coba 5BB. And in this setup, all the grapevines are ungrafted, as I just said. And the tubes with the grapevines were extracted from the ground, and subsequently each tube was opened using an angle grinder, which completely exposed the root system. And this procedure ensured accurate selection and sampling of the rhizosphere soil, which was taken in the very close proximity to the roots. And afterwards, microbiome analyses were performed. And I would like to show you some of the results here on the right side. So we detected some statistical significances. So here on the slide, you can see linear discriminant analysis effect size taxonomic cladogram comparing the bacterial diversity of the 4 different rootstocks, which you can see here on the right. And each ring represents a taxonomic level with the kingdom, phylum, class, order, family, and genus appearing from the center to the outer line of the circle. And each node represents a taxonomic unit found in the dataset. And for each significantly discriminating taxon detected, the corresponding node and branch region for this taxon is colored. As you can see here, the color, for example, for 1,103 Paulson is purple. So all the purple branches are significant different in this rootstock to the other ones. And if the taxon is not significantly different between the rootstocks, the corresponding node is colored yellow. And in total, we found 68 bacterial clades, which were differently distributed among the rootstocks. And the rootstock 1,103, so the purple rootstocks here, showed the highest number of differentially abundant taxa. So these results show us that the rootstock genotype is affecting the composition or the structure of the bacterial community in the rhizosphere, as you can see on the what I showed you on the cladogram. But we don't know why they are different. So we can see that there are differences in the bacteria between the different rootstocks. But as I just said, that the rootstocks are usually used to graft vines, other grapevines on them. We are not sure if we have the same results in the field, because as you might know, a grapevine or perennial plants may root really, really deep. So it's really hard to get a real rhizosphere out in the field. Yeah, so we have to do some more research to use this outcome of this experiment from a practical perspective. And yeah, furthermore, most of the bacteria have still unknown functional capabilities, and also the mechanism of the grapevine recruiting its microorganisms is still unknown. So we have to do further studies, and we want to look at the different rootstocks more further, and we also want to have a look on the different science to graft on in the different grape varieties, and also on the different experimental setups like different fertilization levels or different abiotic factors like water stress or something else. Yeah, so it's a very young project and we want to go on with this. So thank you for your attention. Do you have any questions?
Thank you, Miss Leonie. It was a nice presentation. Is anyone having any questions? Okay, I think there are not. So thank you. Next we can go for video presentation. I will be playing the video and will share you with the screen. And the next presentation is from Dongdong Gyan Academy of Agriculture Sciences, China. Is it visible to everyone?
Our research focuses on the bioactivity and the efficacy of DMDS. And my presentation contains the following three parts. Soil-borne disease and nematodes cause great damage in greenhouse crops and consequently lead to reduction in crop yield. Soil fertilization is an effective technique for control soil-borne pests. Soil-borne pests, including soil fungi, bacteria, nematodes, virus, weeds, and so on. Soil fumigation has been widely used on strawberry, tomato, cucumber, ginger, tobacco, watermelon, and many high-value crops in China. These are different soil fumigation technologies used in China, including non-chemical and chemical fumigation technologies. And we can see the crop yield and plant growth will greatly improve after fumigation. Many kinds of nematodes could cause plant soil-borne disease, such as root-rot nematode, cyst nematode, stem, barber nematode, and so on. Root-rot nematode is widely distributed worldwide. There are four most common species of root-rot nematode. To date, approximately 23 species of the root-rot nematode have been described in China, and 75 non-Chinese species have been reported. New ones are being reported every year. It attacks more than 2,000 kinds of higher plant species and causes great damage to agricultural production. And this is the nematode infection of tomato plants. Chemical treatment is an effective way to control nematodes. Generally, there are two types of nematicides, fumigant and non-fumigant. And these are common non-fumigant nematicides widely used. Also include some biological control agent. Some of the nematicide products were just registered in the past 5 years. And these are common fumigants used worldwide. Soil fumigant usually give excellent control of nematodes in soil. All fumigants have a low molecular weight and occur as gases or liquid. As the fumigant volatilizes, the gas is diffused into the space between the soil particles and kill the nematode.
DMDS is a sulfide-based volatile compound, and it is a natural biopesticide. Anime species are the natural source for DMDS production. And DMDS was first registered in 2010 for pre-plant soil fumigation by Agma, a French company, and now it is commercially available for vegetable crops to control nematodes and other soil-borne pathogens in USA, Turkey, Egypt, Korea, and Mexico. And DMDS is under registration in China. This is the small injection machine for DMDS application in China. The small injection machine can work both in the open field and the small greenhouse in China. And our research results contains 2 bars to bioactivity and the feed efficacy of DMDS. In our laboratory experiment, 3 methods were used to identify the fumigation and the soil fumigation activity of DMDS. The bioactivity of DMDS was compared with 1,3-D, a traditional fumigant for laboratory control. For the direct contact activity, the LC50 of DMDS is 29 milligram per liter, which LC50 of 1,3-D is 18 milligram per liter. It means 1,3-D has a higher direct contact activity than DMDS. The direct contact activity was determined in the water solution, so the activity is seriously affected by the emulsification efficiency of the pesticide. And both DMDS and 1,3-D have excellent fumigation activity. The fumigation activity of DMDS is quite similar to 1,3-D. And for the soil fumigation activity, 1,3-D has a higher activity than DMDS. In our field experiment, 4 doses of DMDS was set in our experiment, ranged from 10 to 80 grams per liter per square meter. The experiment was conducted in greenhouse cucumber and tomato. The root-lot index and the crop yield for determining each treatment. From the results, we can see that the highest root-lot index was observed in untreated control, and all DMDS treatment reduced the root-lot index. And this is the tomato and cucumber root in fumigation and untreated control treatment. It is quite obvious that the plant root in fumigation treatment has root loss. And all DMDS fumigation treatment significantly increased the crop yields compared to the untreated control in both cucumber and tomato field. And in conclusion, in our laboratory study, DMDS shows good bioactivity on nematodes, and the recommended dose for DMDS for feed fumigation is 10 to 20 grams per square meter for controlling root-lot nematodes of short-term crops such as cucumber and tomato. And DMDS is a technically feasible soil fumigant for controlling root-lot nematodes in China. Besides the research on the bioactivity and the efficacy of DMDS, we also carry out study on the combination use of DMDS with other soil fumigants, such as chlorpyrifos 1,3-D and benzoate. And due to the bad smell of DMDS, we also developed a new formulation of DMDS to minimize the bad smell. And that's all for my presentation, and thank you for your listening.
Yeah. Now our next presentation is from Nergis Gürbüz Kolak from İzmir Institute of Technology, Turkey.
Welcome everyone. I'm Nergis Gürbüz Kolak from İzmir Institute of Technology. I graduated from the chemistry department and I completed my graduate education at molecular biology and genetics department. Now I'm working at Plant Science and Technology Application and Research Center as a researcher. Today I'm gonna give a brief talk about plant breeding, both genomics and metabolomics approach in plant breeding. So the first question is, why do we need plant breeding? As you know, world population is dramatically increasing. Nowadays, nearly 8 billion people exist on Earth, and it is estimated that human population will reach almost 10 billion people by the year 2050. This increasing human population is facing us with a serious problem, which is hunger, and the other problem is malnutrition. Unfortunately, today, 800 million people go to bed hungry. Even worse, 15 million people, especially children, die every year because of hunger and malnutrition. What can we do to deal with this bad scenario? We need to improve our agricultural production at least 60 to 70% to feed the growing world population. This brings us to plant breeding. In its simplest form, we should increase the yield in order to increase the agricultural production. But also, hunger is not the only reason of plant breeding studies, of course. The growing human population faces a number of agricultural challenges, including global warming, climate change, decreased soil quality due to pesticides or herbicides, and the number of land races are a problem. And growing industrialization causes reduction of agricultural lands. Also, both abiotic and biotic stresses cause reduction of yield. Besides this, also consumer awareness is changing. Nowadays, people want to eat healthy foods, and we want to get enough nutrition in our daily diet. So, feeding people is important, not only in terms of quality, but also quality is important. Because many chronic and autoimmune diseases are closely related to diet. All these bring us breeding of plants and improvement of new cultures. So, today breeders need to improve important agronomic traits such as yield and develop resistant and adaptive cultivars to climate change. Also, resistant cultivars to abiotic stresses. Besides this, breeders need to satisfy the demands of consumers for nutritional quality or for safe food. Genetic diversity is the most important issue in plant breeding studies. Genetic diversity has been the basis of plant breeding since the earliest days of agriculture. Strategies to achieve these objectives include widening the pool of genetic diversity, to discover novel variation, and to create new growth variation phenomena. Also, understanding the genetic basis of both simple and complex traits and efficient integrations of these variations into new cultivars are important. Plant genomics, in its broadest sense, includes all system approaches to study of model plants and crops, and still remains a key field in the process of improving crops. Plant genomics aims to sequence, characterize, and study the genetic compositions, structure, organizations, functions, and interactions on networks of an entire plant genome. The genomic approaches are useful for accelerating variety development and crop improvement. We can detect QTLs. I will mention QTLs in the next slides. We can detect favorable and unfavorable traits and others. Breeders use genomic tools to develop new cultures. There are two main strategies, which are One of them is conventional breeding, and the other one is micro selection and breeding. First, classical breeding involves interbreeding of individuals selected based on phenotype to develop new cultures. Selection, evaluation, and crossing of useful genotypes require years and several generations. And we can list the disadvantages of conventional breeding as it just relies on the phenotypic selection. Phenotypes, as you know, are affected from environment, and this can be misleading as it is not due to genotype. Also, some measurement errors can be arised when evaluating the qualitative traits such as color. As I said, conventional breeding takes years and many generations, so it's really slow. On the other hand, molecular breeding uses molecular biology and biotechnology techniques to develop new cultures. DNA polymorphism is used to select useful genotypes, which reduces the selection period and number of generations available. Molecular breeding includes two major approaches: microacid selection and genetic transformations or genetic engineering. Genetic engineering applications in plant breeding are very limited because of food safety and environmental effects. So we will focus on microacid selection. However, microacid selection has been widely applied in plant breeding. Compared to conventional breeding, microacid selection provides more efficient screening and early selection. And microacid selection reduces time and thus reduces cost. Moreover, we have chance to select heterozygous plants. Recent advances in next generation sequencing, especially genotyping by sequencing, provide powerful microacid selection tools for plant breeding and crop improvement. Actually, sequencing technologies have brought a revolution in the field of plant genomics. In the first step in GBS, a library is prepared by digesting DNA sample with a restriction enzyme. Then barcodes are added to the sticky ends of restriction site. Each sample has its own unique barcode sequence, and DNA fragments are amplified by PCR and sequenced, and the barcodes are used to assign sequences. The main advantage of GBS is we can simultaneously perform SMP discovery and genotyping. So the next question is, what is SMP? SNPs are single nucleotide polymorphisms. SNP markers represent difference in single nucleotide base among individuals or two DNA sequences. Of course, these differences can be arise from transitions or transversions. SNP markers, of course, they have both advantages and disadvantages. They are highly polymorphic. They present in the simplest form of polymorphism. They are dominant markers. They are suitable for automation, and they are effective to detect polymorphism. These are advantages of SMP markers, but also they have limitations. Startup and marker development prices are really high, and high quality DNA is required, and expensive equipments are needed. These are the limitations of SNPs. So, why do we use SNPs in plant breeding? Mainly, we are using SNP markers to determine the allelic variations. So, we can detect genes and discover QTS to reveal genetic basis of the desired traits for breeding purposes. Here, I will mention about QTS, which are quantitative trait loci. We can define a QTS as a section of DNA that correlates with variations in a phenotype. Usually, the QTR is linked to or contains the genes that control the phenotype. Quantitative traits are not simple, they are complex. The quantitative trait shows continuous variation in population. And QTRs are under the combined effect of many genes, not only one. They show bicurve distribution of phenotypic values in a population and produce a range of phenotypes. That's because most traits of agricultural relevance are progenic. So, the next question is, what is the purpose of QTR studies in plant breeding? The purpose is to localize chromosomal regions that significantly affect the variation of quantitative traits in the population. This brings us to QTR mapping. When we know the locations of QTRs on the chromosomes, we can integrate favorable QTRs, favorable regions, which are responsible for the desired traits into a new variant. Projectively, QTRs are being used to identify the location of genes and major and minor alleles that are responsible for identifying traits, and to investigate their effects in crops and exploration for crop improvement programs. So far, many of genes have been mapped, cloned, and their function identified in crops using QTR. Actually, QTR mapping is not a new technique, but still remains a powerful technique to study complex agronomical traits. In this slide, I just want to remind a few definitions. So far, we've talked about plant genomics and functional genomics. So, as you know, genomics is the analyzing and sequencing of an organism's entire genetic makeup, and it provides molecular insights to understand the function, structure, and the inheritance of the genetic makeup, and improve the complex traits. And functional genomics studies function, expression profiles, and regulation of genes, interaction between genes, and the responses they exhibit in phenotype. Functional genomics not only studies data that are generated from genome biomolecular virology techniques, but also involves metabolomics. So, we are going to talk about a little bit metabolomics. Metabolomics is an omics approach that has gained popularity over the past few decades and refers to systemic identification and quantification of the small molecule metabolic products of a biological system at a specific point in time. Metabolomics provides full and global comparison of the differences between cell types, tissues, organs, and whole organisms. So we can find the normal aspects of gene function, physiology, and metabolism for many future research goals. Metabolomics or metabolic profiling provide us to discover new pathways. Metabolic markers have become an important tool to uncover and investigate the various biological complex pathways responsible for distinct phenotypes. Metabolomics decreases the gap between the phenotype and the genotype, and this approach connects the metabolome and genome and provides important insights into genetic function and investigates phenotypic variation via metabolic profiling and comprehensive gene expression analysis. To understand the metabolic networks that reflect complex developmental processes, metabolomics-based quantitative trait loci studies are important for improving the quality and the performance of cultivars. In addition, results obtained from MQTR studies contribute to a deeper understanding of quantitative and functional genetics. There are mainly two approaches in metabolomics studies. The first one is targeted metabolomics. In targeted metabolomics, we are quantifying specific target molecule or molecules. For example, we are interested in improving flow. So it is important to quantify volatile compounds, organic acids, and sugars. which contribute to flower. So we need to evaluate the variation of these metabolites in a population to improve the flower. The other approach is untargeted metabolites. Here we don't have a specific target or specific target molecules. We reveal global changes in all metabolites. For example, we can compare the changes in metabolite concentration under stress conditions. Up to now, we've talked about application of genomics and metabolomics in plant breeding. Now, for an example, I want to talk about a study we've done in our lab. We worked on tomato, and up to now, in literature, tomato breeding studies have focused on to improve yield, fruit quality, resistance to stress, nutritional value, and medicinal compounds content. First, I want to talk about why we are working on tomato and the importance of tomato. Tomato belongs to Solanaceae family and Solanum genus, and the species is Solanum lycopersicum. It's an diploid organism and chromosome number is 12. What species of tomato grow in a wide range of habitats from sea level and from arid to rain climates. It's an economically important plant. It is used to make paste, soup, ketchup, juice, and etc. In this chart, we are seeing the top producers of tomato. China is the top producer. India, United States, and Turkey follow China. And also, my country, Turkey, is an important producer and exporter of tomato. Besides its economic importance, as you see in the table, it is also important in terms of nutrition. It is rich in minerals and vitamins, especially vitamin C, and also some antioxidant molecules like beta-carotene, and especially lycopene. Moreover, tomato is used as a model organism because it has a relatively short life cycle. It can be grown in different conditions, and this allows us to study changes and adaptability to acute stress conditions. It has high cell fertility and homozygosity, and has photoperiod insensitivity. That means this provides the ability to produce seeds and flower in any condition of daylight. This ability to be propagated asexually by grafting, and propagation in tissue culture is easy to transform and regenerate. It has simple and relatively small genome, and its whole genome was sequenced. And these are all advantages of tomato. Thus, breeding of tomato becomes an important issue. Tomato breeding started at the end of the 19th century, and the next century, public institutes and private companies were involved in tomato breeding. The first hybrid tomato released in the middle of the 1900s and named single crush. Now, private companies dominate the market. They should be competitive and innovative to continue to dominate the market. So they should find new genetic resources to develop new products. White species represent high genetic variation, so they can be new resources for the development of new cultures. During the tomato breeding studies for 200 years, genetic variation among tomato varieties because during domestication, selection of tomato was done on a single plant basis and with small numbers of plants in the existing germplasm, which results in decreased genetic variation. In addition to this, genetic variation tends to decrease in ingredient species even without selection. So, we need to find new resources and we need to expand genetic variation. For this purpose, what species are good choices? Now I want to briefly talk about our study a little bit. We use Solanum lycopersicum as cultivated parent and Solanum pinguinifolium as the white relative of tomato. We created BC2, F6, population from these two parents. For this purpose, genotyping of tomato was done by using TBS approach. We evaluated phenotypic and agronomic traits such as pH, fruit weight, diameter, external and internal color, local number, bulb thickness, firmness, fruit shape, stem scale, and soluble solid content. And we performed targeted metabolomics for the quantification of sugars, organic acids, beta acids, vitamins, carotenoids, glutathione, antioxidant molecules, phenolic acids, and volatile compounds. Finally, we identified critical regions for agronomic traits, antioxidant and flower-related and nutritional traits. As a result of TBS approach, we detected more than 3,000 FMPs, which were located all of the tomato chromosomes. And we published the TBS results. This study is the first report of high-throughput SMP discovery using TBS tomato. That was the first study in which QTLs for fruit-filtered traits were identified by constructing a high-density SMP-based distal map using developed IBR population that carries integrations from Solomon folium genome. And also, we published another study for antioxidant characters in tomato. In the study, white tomato, which is Solomon folium, was assessed for its potential to improve antioxidant content. This white species was found to be a good source of alleles for increasing, especially beta-carotene, lycopene, vitamin C, and vitamin E contents in cultivated tomato. All these molecules are antioxidant molecules. And we identified 64 criteria for the antioxidant traits. In another study, we identified criteria for tomato flower compounds. Solanum pimpinellifolium, in the study, Solanum pimpinellifolium was found to harm alleles that could be used to increase glucose and fructose content and adjust acidity by altering medicinal levels. And we measured sugars and organic acids, and 85 criteria were identified for the flow-related traits in this paper. Also, we published our methods, which were used to analyze antioxidant and flow-related metabolites. And now we are preparing another paper from this project, which is related to nutritional characters in tomato. And this is the end of.
So by this we came to end of the day 2 webinar. I would like to thank all the attendees and participants of today's webinar. Hope you enjoyed the webinar and gained knowledge from the sessions. And we will be glad if you could join us in our next webinar, which will be scheduled, and the dates will be informed to you by mail. We will be very happy if you could share your opinion or feedback with us. If you have any queries, you can mail us to the concerned program manager. Thank you. Have a nice day.
Hey, Jessica, if I could for a minute, it's Damon here. Sravani just sent me some documentation, and there are edits that need to be done. I messaged her and I let her know. So if you could get her to deal with that for me, I'd greatly appreciate it. And it's just clarification as to how you guys have me listed in that document. So thank you.
Thank you, doctor.
Not a doctor.
Oh, sorry, Mr. Damon. We will
Thank
be
you.
happy. We'll be happy if you could join us on the next webinar.
I will consider it. So thank you very much, Jessica. And yeah, if you could get Sravani to edit what was put there, I'd appreciate that.
Yeah, definitely.
It's a mistake. mistake on your end. So thank you.
Okay, thank you. Thank you. Have a nice day.